In the shallow coastal waters of the world's temperate and tropical seas, the seafloor appears calm at first glance, a patchwork of sand, coral rubble, and drifting shadows cast by sunlight filtering through moving water. Yet beneath that quiet surface lies a biological reality that is far more ruthless than it appears. Every inch of this landscape is contested by predators, grazers, scavengers, and microscopic opportunists that constantly test the limits of survival. Here, damage is not an occasional event, but an everyday certainty. Fish bite, crabs crush, storms scatter bodies across the seabed, and hungry mouths remove limbs without hesitation. In most forms of life, losing the majority of one's body would end the story almost instantly. But scattered across this battlefield are animals that treat catastrophic injury not as an ending, but as a beginning. Among them, the starfish stands as one of the ocean's most astonishing demonstrations of biological resilience, an organism whose relationship with destruction has been rewritten by evolution itself. A starfish resting on the seabed does not appear particularly formidable. Its soft body spreads outward into 5 arms that radiate from a central disc, giving it a shape that seems almost decorative against the sand or coral. Colors range from muted browns and grays to vivid oranges, reds and purples that glow against the blue water above. Tube feet ripple slowly along the underside of each arm, tiny, flexible structures driven by an internal hydraulic system that allows the animal to glide across rock and sediment with patient determination. Yet beneath this seemingly simple design lies a body plan so radically different from most animals that it allows for feats of survival that verge on the unbelievable. The starfish belongs to a group of animals known as echinoderms, a lineage that also includes sea urchins, brittle stars, and sea cucumbers. These animals share a structural feature that immediately sets them apart from creatures with bones, muscles, and centralized organs. Instead of relying on a rigid internal skeleton and a compact arrangement of vital organs, the starfish spreads its essential functions across its entire body. Its nervous system forms a circular nerve ring that surrounds the mouth in the center of the disc, from which radial nerves extend outward along each arm, like the spokes of a wheel. Rather than concentrating control in a single brain, the starfish distributes processing power across its limbs. Each arm contains sensory structures and neural networks capable of coordinating movement and responding to environmental signals. In effect, the animal operates more like a biological network than a centralized organism. This distributed design becomes most remarkable when disaster strikes. Across coral reefs and rocky coasts, starfish frequently encounter predators that treat their arms as convenient meals. Fish tear pieces away. Crabs pinch and sever limbs with powerful claws. Octopuses pull them apart with quiet efficiency. Storms and currents can also rip starfish from rocks and scatter fragments across the seabed. In most animals, such catastrophic damage would quickly disrupt circulation, nerve function, and the coordinated activity of organs necessary for survival. But the starfish possesses a remarkable biological capability that transforms trauma into opportunity. Instead of collapsing when its body is torn apart, it begins rebuilding. The process begins almost immediately after injury occurs. When an arm is lost, specialized cells near the wound rapidly seal exposed tissue, preventing infection and limiting further structural damage. The water vascular system, the internal network that powers the tube feet, adjusts pressure throughout the body so that the remaining limbs continue functioning. Meanwhile, cells near the injury site undergo a remarkable transformation. Some revert to a more primitive, flexible state known as de-differentiation, shedding their specialized roles in order to become building material for new structures. From this pool of adaptable cells, regeneration begins. Over the following days and weeks, a small swelling forms at the point where the arm was severed. At first, it appears almost insignificant, little more than a soft mound of tissue. But beneath the surface, a complex orchestration of genetic signals guides the reconstruction of lost anatomy. New muscles form, connective tissues align, and the radial nerve begins extending outward once again. Gradually, the swelling lengthens into a developing limb, growing tube feet, skeletal elements, and sensory structures as it expands. Over time, the arm regains its full shape and function, restoring the starfish to its original form. What makes this ability extraordinary is not merely the regrowth of a lost limb, but the fact that the process can occur in reverse. In certain species, the regeneration process extends beyond replacing parts of an existing body. Instead, an entirely new individual can emerge from what was once only a fragment. This phenomenon is known as fission, a reproductive strategy that transforms destruction into duplication. Imagine a starfish crawling slowly across a reef surface, its tube feet probing the rock for chemical traces of food. Suddenly, a predator strikes, tearing one arm away from the central disc. The predator may consume part of the limb, but if even a portion of that arm remains intact and contains a small segment of the central disc where the nerve ring once connected, the detached fragment does not simply decay. It begins rebuilding itself. Within that severed arm, the same regenerative machinery activates once again. Cells reorganize, tissues reform, and a new central disc gradually begins to take shape near the base of the limb. The radial nerve that once extended outward now becomes the foundation for reconstructing an entirely new nervous system. Over weeks or months, 4 additional arms emerge around the growing disc, transforming what was once a fragment into a complete organism. In effect, the arm becomes the seed of a new starfish. This process creates what might be described as a multi-core organism, one whose functional units possess enough independence to rebuild the whole if given the chance. Instead of relying on a single fragile control center, the starfish spreads the blueprint for its existence throughout its body. Every arm contains the biological instructions necessary to reconstruct the entire animal, provided the conditions are right. From an evolutionary perspective, this ability offers profound advantages in environments where injury is unavoidable. The seafloor is a landscape defined by constant interaction between predators and prey. For many animals, losing a limb represents a devastating setback that compromises mobility, feeding, or defense. For the starfish, however, injury becomes a manageable inconvenience. A lost arm can be replaced, and in some cases it may even produce a new individual, effectively turning predation attempts into accidental acts of reproduction. The ecological consequences of this ability ripple through entire marine communities. Starfish are often powerful predators themselves, capable of feeding on shellfish, coral, and other invertebrates by prying open shells or everting their stomachs outside the body to digest prey externally. Because they can recover from severe injuries that would kill other predators, their populations can remain stable even under heavy predation pressure. This resilience allows them to maintain their role as regulators within the ecosystem, controlling populations of mussels, clams, and other species that might otherwise dominate the seafloor. Regeneration also shapes the starfish's behavior. Unlike animals that must avoid injury at all costs, starfish can tolerate risk during feeding and exploration. They wedge their arms into narrow crevices, pry apart tightly closed shells, and engage prey that might fight back. Even if a limb is damaged in the struggle, the long-term consequences are relatively minor compared to the rewards of securing a meal. Scientists studying this remarkable ability have discovered that the genetic mechanisms underlying starfish regeneration are deeply complex and finely controlled. Genes responsible for embryonic development reactivate during regeneration, effectively replaying aspects of the organism's earliest growth stages. Signaling pathways guide cells to become specific tissues, ensuring that muscles, nerves, and skeletal elements form in the correct positions. The process must remain precisely balanced, since uncontrolled growth could lead to malformed structures rather than functional limbs. What fascinates researchers even more is the potential insight this process provides into broader biological questions. Regeneration remains limited in most vertebrates, including humans, where severe injuries typically result in permanent loss of tissue rather than reconstruction. By studying the cellular signals and genetic programs that allow starfish to rebuild complex body structures, scientists hope to better understand why regenerative capacity differs so dramatically across the animal kingdom. Yet within the ocean itself, the starfish's regenerative ability serves a far more immediate purpose. It is a survival strategy forged by millions of years of evolutionary experimentation in environments where damage is unavoidable. Predators attack, storms break bodies apart, and the seafloor constantly rearranges itself through currents and geological movement. In such conditions, resilience becomes as important as strength or speed. The result is an organism that defies the typical boundaries of life and injury. A starfish can lose most of its body and still continue living. A detached arm can reorganize itself into an entirely new animal. A predator that consumes part of a starfish may unknowingly leave behind the beginnings of another one. Across the reefs and rocky coastlines of the world, this process repeats quietly and constantly. Limbs are torn away, fragments settle onto the seabed, and regeneration begins once again beneath the drifting shadows of passing fish. To an observer watching from above, the scene appears unchanged. But within the tissues of these strange animals, destruction has been rewritten as transformation. In the relentless contest for survival beneath the ocean's surface, few creatures demonstrate the concept of biological persistence more clearly than the starfish. Where most animals see the end of life in catastrophic injury, the starfish sees only the beginning of reconstruction. Through regeneration and fission, it transforms loss into multiplication, injury into opportunity, and fragments into entire living beings that crawl slowly across the seabed, ready to repeat the cycle once again. Along the cold, shadowed floor of the North Atlantic, the seabed stretches across vast fields of rock, broken shell, and drifting sediment that shifts slowly under the influence of tides. The water above moves in long, deliberate currents that carry microscopic life through the dim blue light filtering from the surface. Among the crevices of boulders and the narrow tunnels beneath ledges, a creature moves with deliberate patience, its armored body blending into the rough textures of the seafloor. The American lobster emerges from its shelter at dusk, its antennae extending forward like sensory cables that sweep the water ahead. Every movement of those antennae gathers information from the surrounding environment, detecting faint chemical trails left behind by prey, rivals, or predators. To most animals that share this ecosystem, life follows a familiar biological rhythm: youth, maturity, decline, and eventually death, as age erodes the systems that once sustained them. But within the body of the lobster, evolution has produced a peculiar biological anomaly that challenges this pattern in a remarkable way. The lobster's body appears built for endurance. Its thick exoskeleton forms a rigid shell that protects the delicate tissues within, while powerful claws allow it to pry open shells or defend itself from intruders. Beneath that armor lies a physiology shaped by millions of years in cold, oxygen-rich water. The lobster's circulatory system moves hemolymph through its body, delivering oxygen and nutrients to muscles that allow the animal to crawl across the seabed with surprising strength. Its gills extract oxygen from water flowing through chambers beneath the carapace, while complex sensory organs detect vibrations, chemical cues, and subtle changes in water pressure. Yet the most unusual feature of the lobster lies not in its claws or armor, but within the microscopic machinery of its cells. In most animals, aging is not merely the passage of time, but the gradual failure of a system designed with built-in limits. Within every cell, strands of genetic material contain instructions for building and maintaining the body. At the ends of these strands sit protective caps known as telomeres, structures that function like the plastic tips at the ends of shoelaces. Each time a cell divides, those caps shorten slightly. Over the course of thousands of divisions, the telomeres become too short to protect the genetic material effectively. At that point, the cell either stops dividing or begins to malfunction. This gradual shortening acts as a biological clock that limits how long tissues can renew themselves. In humans and most other animals, it is one of the fundamental mechanisms behind aging. The lobster, however, appears to operate under a different set of rules. Deep within its cells is an enzyme known as telomerase, a molecular tool capable of rebuilding those protective telomere caps after each cell division. In many animals, telomerase is active only during early development, after which its activity diminishes as the organism matures. Lobsters, by contrast, continue producing this enzyme throughout their lives. As a result, their cells maintain the ability to divide and repair themselves without the gradual erosion that typically accompanies aging. In effect, the cellular clock that drives senescence in most organisms never fully activates within the lobster's tissues. This unusual biological feature produces a remarkable consequence. Unlike many animals whose metabolism slows and tissues degrade over time, lobsters retain their physiological vitality for decades. A mature lobster is not an elderly version of its younger self in the way a mammal might be. Instead, it often becomes stronger and more capable as it grows larger. Its claws thicken, its shell becomes more formidable, and its reproductive capacity remains active far into what would be considered old age in other species. The lobster's body continues functioning with the efficiency of a mature adult, rather than sliding into gradual decline. Yet the lobster's life is not entirely free from limits. Its unusual relationship with aging comes with a different challenge, one tied to the rigid armor that protects it. Unlike animals with flexible skin or internal skeletons, the lobster cannot simply grow larger within its existing shell. The exoskeleton is a fixed structure that must be shed and replaced periodically through a process known as molting. During molting, the lobster withdraws its body from the old shell and expands within a newly formed soft one that gradually hardens as minerals accumulate within the tissue. For a young lobster, this process occurs frequently. Juveniles molt many times within a single year, each molt allowing rapid growth as the animal increases in size. With each shedding of the shell, the lobster emerges slightly larger and stronger than before. As it matures, the frequency of molting decreases, but the process remains essential throughout its life. Each molt requires an extraordinary amount of energy. The lobster must absorb minerals from its old shell, withdraw from it, and inflate its body within the new exoskeleton before the surrounding water pressure collapses the soft tissue. The vulnerability of this moment cannot be overstated. When a lobster molts, it becomes temporarily defenseless. Its new shell remains soft for several days, leaving the animal exposed to predators that might otherwise struggle against its hardened armor. During this time, the lobster retreats into narrow crevices or burrows beneath rocks, relying on concealment rather than strength for survival. Once the new shell hardens, it emerges once again to resume its role as both predator and scavenger on the seafloor. For many years, scientists believed lobsters might effectively live forever because of their cellular biology. Their ability to maintain telomere length and sustain cell division suggested that the usual molecular triggers of aging were largely absent. However, careful observation of older individuals revealed a different limitation. As lobsters grow larger, the energy required to complete each molt increases dramatically. The exoskeleton becomes thicker and more mineralized, while the internal tissues must expand to fill an ever greater volume. Eventually, a point arrives when the lobster's metabolism can no longer generate enough energy to successfully complete the molting process. The animal may begin the cycle, but lack the strength required to fully escape the old shell. In other cases, the stress of molting becomes so intense that the lobster's physiological systems fail during the process. Instead of dying from the slow deterioration typical of aging, the lobster reaches a mechanical limit imposed by its own growth. This unusual constraint creates a fascinating paradox. The lobster does not die because its cells wear out over time, as happens in many animals. Instead, it dies because it grows too large to maintain the demanding process that allows it to keep growing. Its body never activates a biological off switch that signals old age. Instead, the accumulation of size itself becomes the challenge that ultimately ends its life. In the wild, few lobsters ever approach this theoretical limit. The ocean is not a place that rewards longevity without challenge. Predators patrol the seabed, including large fish, seals, and octopuses that view lobsters as valuable prey. Fishing activity also removes enormous numbers of individuals long before they reach extreme sizes. Yet occasionally, fishermen capture specimens that hint at what a lobster might become if left undisturbed for many decades. These individuals can grow to remarkable proportions, their claws massive, and their shells bearing the scars of countless encounters with predators and rivals. Observing such animals provides insight into the strange consequences of their biology. A large lobster may be many decades old, yet its tissues remain robust, its reproductive organs active, and its behavior vigorous. In this sense, the lobster represents one of the clearest examples of what scientists call negligible senescence, a condition in which the typical markers of aging remain minimal even after long periods of time. This biological resilience influences the lobster's ecological role as well. Large individuals become dominant predators within their territory, capable of cracking open the shells of clams, mussels, and other invertebrates that form the foundation of coastal ecosystems. By controlling populations of these organisms, lobsters help shape the structure of benthic communities. Their feeding activity alters the balance of species on the seabed, preventing any single group from overwhelming the habitat. The lobster's ability to maintain vitality over long periods also affects its reproductive strategy. Female lobsters carry thousands of fertilized eggs beneath their tails, protecting them until they hatch into tiny larvae that drift into the plankton. Because older individuals remain reproductively active, they continue contributing to the next generation long after many other animals would have lost that capability. In this way, the lobster's unusual biology allows experienced individuals to remain productive members of the population for decades. From a scientific perspective, the lobster's relationship with aging raises profound questions about the nature of biological limits. If the molecular triggers of senescence can be suppressed through mechanisms like continuous telomerase activity, it suggests that aging is not an unavoidable feature of life, but rather one possible evolutionary strategy among many. Different species have adopted different solutions to the challenges of survival, reproduction, and resource allocation. The lobster's solution appears to involve prioritizing long-term cellular maintenance while accepting the physical constraints imposed by its exoskeleton. Instead of allowing tissues to degrade gradually, it invests heavily in molecular repair systems that keep cells functioning efficiently. The trade-off emerges later, when the cost of molting eventually becomes too great to sustain. Across the rocky sea floor of the North Atlantic, the lobster continues its slow, nightly patrols, antennae tracing invisible chemical trails through the dark water. Each step carries it across a landscape shaped by currents, predators, and the constant search for food. Within its cells, the molecular machinery that rebuilds telomeres continues operating without interruption, quietly maintaining the integrity of its genetic code. The animal itself remains unaware of the extraordinary biology unfolding within its tissues. It simply lives as lobsters have lived for millions of years, guided by instincts shaped by evolution. Yet to those who study the ocean's creatures, the lobster represents something far more intriguing than a crustacean scavenger crawling across the seabed. It is a biological anomaly, an organism whose cellular machinery refuses to obey the usual limits imposed by time. In a world where most life follows a predictable path from growth to decline, the lobster stands as a reminder that evolution sometimes produces solutions that appear almost like glitches in the code of life itself. Along the slopes of coral reefs and across the quiet plains of sandy seabeds, an unusual creature lies scattered across the ocean floor like discarded fragments of living leather. To a casual observer, it might appear inert, almost plant-like, a soft, elongated body, resting motionless among the rubble of shells and coral fragments. Yet the sea cucumber is neither fragile nor passive. It is one of the strangest animals the ocean has produced, a creature whose survival depends not on armor, speed, or venom, but on the remarkable ability to alter the physical nature of its own body. Within seconds, the tissues of a sea cucumber can transform from the firmness of cartilage into something resembling liquid. Structures that once held shape dissolve into a flexible, flowing mass capable of squeezing through impossibly narrow spaces before solidifying once again. The environment in which this ability evolved is one defined by relentless pressure. The seafloor is not the quiet desert it might appear from above. It is a crowded landscape of predators and scavengers constantly searching for food. Fish glide just above the sand, their eyes scanning for movement. Crabs patrol with raised claws, probing beneath stones for hidden prey. Starfish move slowly but persistently across the seabed, capable of enveloping animals that cannot escape quickly enough. Even the currents themselves pose a threat, shifting sediments and exposing animals that rely on concealment. In such a place, an organism that cannot run or fight must rely on a different kind of strategy. The sea cucumber belongs to the echinoderms, the same evolutionary lineage that produced starfish and sea urchins. Yet its body plan has diverged dramatically from its relatives. Instead of rigid spines or clearly defined arms, the sea cucumber has evolved into a flexible cylinder whose outer surface appears soft and unremarkable. Beneath that exterior lies a complex internal structure supported by a unique form of connective tissue known as mutable collagenous tissue. This tissue is the key to the sea cucumber's most extraordinary survival mechanism. In most animals, connective tissue behaves in predictable ways. Tendons, ligaments, and cartilage possess fixed mechanical properties determined by the arrangement of collagen fibers and the biochemical bonds that hold them together. Muscles may contract or relax, but the structural framework supporting them remains relatively constant. The sea cucumber's connective tissue operates under a different principle entirely. Through precise chemical signals controlled by its nervous system, the animal can alter the stiffness of this tissue almost instantly. When the collagen fibers within the tissue are tightly bound together, the body becomes firm and supportive, allowing the sea cucumber to maintain its shape while crawling slowly across the seabed using rows of tube feet along its underside. When those bonds are chemically weakened, however, the structure collapses into a soft, pliable state. The entire body becomes dramatically more flexible, capable of bending, compressing, and flowing into spaces that would seem impossible for a solid organism. The transformation occurs through the manipulation of microscopic crosslinks between collagen fibers. Specialized cells release proteins that either strengthen or weaken these bonds. When strengthening proteins dominate, the fibers lock together like reinforced cables, producing a rigid structure that can withstand pressure or resist deformation. When weakening proteins are released, those bonds dissolve, allowing the fibers to slide freely past one another. The result is a rapid transition from structural rigidity to near fluidity. This property is sometimes referred to as catch collagen, a name that reflects its ability to hold tension for long periods without constant muscular effort, while also shifting quickly between states of stiffness and softness. For the sea cucumber, this capability is not merely an interesting quirk of physiology. It is a fundamental survival strategy. Imagine a predator approaching across the seabed, perhaps a large fish investigating the slow movements of a sea cucumber feeding on organic particles in the sediment. The sea cucumber senses vibrations through its body wall and subtle chemical signals in the surrounding water. It cannot swim away or sprint across the sea floor. Instead, it employs a different tactic. Within moments, the connective tissue throughout its body softens dramatically. Muscles contract, compressing the body into a shape that can slip into the smallest crevices within nearby rocks or coral. What follows looks less like movement and more like transformation. The sea cucumber elongates and flattens simultaneously, its body bending around edges and sliding into spaces far narrower than its resting form would suggest. Sections of the body compress into folds that appear almost fluid as they squeeze between fragments of coral rubble. Within seconds, the animal disappears into a crack barely wide enough for a finger, leaving behind only a faint disturbance in the surrounding sediment. Once safely concealed within the crevice, the biochemical signals reverse. Proteins that strengthen collagen bonds are released once again, restoring rigidity to the connective tissue. The body solidifies in place, conforming perfectly to the contours of the hiding space. What moments ago behaved like liquid now becomes firm again, anchoring the animal securely within its refuge. This remarkable transformation allows the sea cucumber to exploit habitats inaccessible to many predators. Narrow crevices, porous coral structures, and tangled rubble fields become safe havens where larger animals cannot reach. The ability to shift between structural states means the sea cucumber can adapt its body to the environment rather than being constrained by a fixed shape. Yet the benefits of mutable collagen extend beyond escape from predators. They also influence how the sea cucumber feeds and interacts with the ecosystem. Most species are deposit feeders, consuming sediments rich in organic matter that accumulate on the seafloor. As the animal crawls slowly across the substrate, tentacle-like feeding structures around its mouth gather sand and detritus, transferring the material into the digestive tract. Within that tract, Microorganisms and decomposing organic particles are extracted as nutrients before the cleaned sediment is expelled behind the animal. This constant processing of seabed material makes sea cucumbers important ecological engineers. By ingesting and reworking sediments, they influence nutrient cycling and oxygen penetration within the substrate. Their feeding activity can alter the distribution of microbial communities and help maintain the health of coral reef ecosystems by preventing the buildup of decaying organic matter. Mutable collagen supports this lifestyle by allowing the body to adapt to uneven terrain while maintaining stability during feeding. When crawling across irregular surfaces, the connective tissue stiffens enough to support muscular movement. When navigating complex coral structures, it softens slightly, allowing the body to conform to the landscape and maintain contact with the substrate. In deeper ocean environments, where light never reaches the seabed and pressure increases dramatically with depth, similar principles apply. Sea cucumbers inhabiting abyssal plains often possess extremely flexible bodies that enable them to traverse soft sediments or squeeze into protective depressions created by geological activity. Some species even develop transparent or gelatinous bodies that enhance their ability to compress and expand within the harsh conditions of the deep sea. The versatility of mutable collagen has also inspired intense scientific interest beyond marine biology. Researchers studying this tissue hope to understand how organisms can dynamically alter the mechanical properties of their bodies without sacrificing structural integrity. The ability to switch rapidly between rigidity and flexibility could one day influence the design of new materials for medicine, engineering, or robotics. Yet for the sea cucumber itself, the significance of this adaptation lies simply in survival. Its body is not designed for speed or aggression. Instead, it embodies a different evolutionary philosophy: adaptability through transformation. By manipulating the microscopic architecture of its connective tissue, the sea cucumber turns its entire body into a tool capable of responding instantly to environmental challenges. The ocean floor is full of creatures that rely on armor, venom, or camouflage to survive. The sea cucumber's strategy is more subtle. When danger approaches, it does not confront the threat or flee across open ground. Instead, it becomes something else entirely, shifting from a solid organism into a flexible mass capable of slipping through the smallest openings in the reef. Moments later, hidden safely within the darkness of a crevice, the transformation reverses. The connective tissue tightens, the body regains its firmness, and the animal remains motionless until the threat has passed. Eventually, when the seabed quiets once again, the sea cucumber emerges slowly from its shelter, its body firm and stable as it resumes the patient work of feeding across the ocean floor. In a world where survival often depends on strength or speed, the sea cucumber demonstrates a different kind of resilience. Its defense lies not in resisting the forces around it, but in changing itself to meet them. By turning solidity into softness and softness back into solidity, it becomes one of the ocean's most unusual masters of transformation. An animal that survives not by holding its shape, but by abandoning it whenever the moment demands. Far out in the open ocean, where the continental shelves fall away and the seafloor plunges into immense blue depths, the surface often appears calm and empty. Wind ripples the water, sunlight dances across waves, and seabirds glide overhead searching for fish. But beneath that tranquil surface lies one of the most extreme vertical landscapes on Earth. As the ocean deepens, sunlight fades, temperature drops, and pressure increases relentlessly with every descending meter. At depths where human-built machines struggle to survive, an enormous predator moves with deliberate confidence through the darkness. The sperm whale is not merely visiting the deep ocean, it is built to dominate it. At the surface, the sperm whale appears almost leisurely as it floats horizontally in the water, its immense rectangular head resting just beneath the waves. A plume of mist bursts upward from its blowhole as it exhales, sending a diagonal spray into the air before inhaling again in preparation for another dive. The animal's skin is wrinkled and scarred from encounters with rivals and prey, marked by pale lines that record decades of life in the ocean's hidden battlefields. Beneath this weathered exterior lies a body engineered for one of the most extreme journeys any mammal regularly performs. Within minutes, the whale will abandon the sunlight entirely, descending thousands of meters into a realm where pressure could crush most animals into lifeless shapes. The deep ocean is defined by pressure. With every 10 meters of descent, the weight of water above increases significantly. By the time an organism reaches a depth of 3,000 meters, the pressure pressing inward on its body is immense, equivalent to thousands of kilograms bearing down on every square centimeter. For animals lacking specialized adaptations, such conditions would collapse airspaces, distort organs, and disrupt circulation. The deeper one descends, the greater the force that attempts to compress the body inward. Yet the sperm whale routinely travels to these depths in search of prey. Its primary targets are giant squid and other deep-dwelling cephalopods that inhabit the twilight and midnight zones of the ocean. These creatures drift far below the reach of sunlight, where the water is cold and the environment remains in perpetual darkness. To reach them, the sperm whale must undertake dives that can last well over an hour while traveling through vertical distances greater than most mountains are tall. The secret to surviving this descent begins with the whale's remarkable approach to breathing. Like all mammals, sperm whales rely on lungs to extract oxygen from the air, but unlike terrestrial mammals, they cannot continue breathing once submerged. Every dive begins with a careful preparation at the surface, during which the whale takes a series of powerful breaths that replenish oxygen stores throughout its body. These breaths do not merely fill the lungs, they saturate the bloodstream and muscles with oxygen-carrying molecules, particularly hemoglobin and myoglobin, which store oxygen in concentrations far higher than those found in land animals. Once fully prepared, the whale arches its enormous tail above the water and begins its descent. Within minutes, it is passing through layers of water, where sunlight fades from bright blue into dim indigo. Schools of fish scatter as the whale glides silently downward, propelled by slow, powerful movements of its tail flukes. The descent continues through depths where many marine mammals would already be approaching their physiological limits. Yet for the sperm whale, the most extraordinary adaptations are only beginning to take effect. The challenge of deep diving is not merely the lack of oxygen, but the crushing force of pressure acting upon any air-filled space within the body. Air compresses dramatically as pressure increases. If a diver were to descend thousands of meters with air trapped in rigid cavities, the collapsing volume could damage tissues and disrupt vital systems. For most mammals, the lungs represent a particularly dangerous vulnerability under these conditions. The sperm whale solves this problem through an adaptation that at first seems counterintuitive. Instead of resisting the compression of its lungs, the whale allows them to collapse deliberately. As the animal descends deeper into the ocean, the increasing pressure compresses the air within the lungs until the delicate air sacs collapse inward. This process effectively removes air from the gas exchange surfaces of the lungs, preventing further absorption of nitrogen into the bloodstream and reducing the risk of decompression-related injuries. To accommodate this transformation, the whale's ribcage must be able to change shape without breaking. The ribs of a sperm whale are not rigidly fixed in the same manner as those of many land mammals. Instead, they are connected through flexible cartilage that allows the entire ribcage to fold inward under pressure. As the lungs compress, the ribcage follows, bending inward rather than resisting the force. This flexibility allows the whale's chest cavity to adjust dynamically as the surrounding pressure increases. The effect is similar to a pressure tank that adapts rather than fractures. While a rigid structure might shatter under such immense compression, the sperm whale's body simply reshapes itself to accommodate the environment. The lungs shrink, the ribcage folds inward, and the air that once filled the chest becomes compressed into smaller spaces within the respiratory system. Meanwhile, oxygen stored within the blood and muscles continues fueling the whale's metabolism during the descent. As the whale moves deeper, its circulatory system undergoes further adjustments. Blood flow is redirected toward the organs that require oxygen most urgently, particularly the brain and heart. Peripheral tissues reduce their oxygen consumption, relying on stored energy reserves rather than continuous circulation. This redistribution allows the whale to extend its dive duration far beyond what would be possible if every part of the body demanded equal oxygen supply. In the darkness of the deep ocean, vision becomes nearly useless. Here, the sperm whale relies on one of the most powerful biological sonar systems ever evolved. Within its massive head lies an organ known as the spermaceti complex, a structure filled with waxy fluid that helps focus and project intense bursts of sound into the surrounding water. Each click produced by the whale travels outward as a concentrated acoustic beam. When these sound waves encounter objects in the darkness, they bounce back as echoes that the whale's brain interprets with extraordinary precision. Through this system, the whale constructs a detailed acoustic map of its environment. Squid moving through the darkness become detectable not through sight, but through subtle echoes returning from the whale's sonar pulses. The clicks grow louder and more rapid as the whale closes in on its target, eventually forming a rapid sequence of sounds known as a buzz that guides the final moments of the hunt. The prey encountered at these depths are often formidable themselves. Giant squid possess powerful tentacles lined with gripping suckers capable of leaving scars on the whale's skin. Evidence of these battles is visible on many sperm whales, whose bodies bear circular marks left by the suckers of squid attempting to escape capture. These encounters unfold in total darkness, guided only by sound and instinct. When the whale captures its prey, it swallows the squid whole before beginning the long ascent back toward the surface. During this ascent, the adaptations that protected the whale during descent continue to function. The compressed lungs gradually reinflate as pressure decreases, while nitrogen absorbed during the dive remains limited due to the earlier collapse of the lungs' air sacs. This mechanism reduces the risk of nitrogen bubbles forming within the bloodstream, a condition known in human divers as decompression sickness. Eventually, after perhaps an hour or more beneath the surface, the whale returns to the world of sunlight. Its massive head breaks through the water, and a powerful exhalation sends a plume of vapor into the air. Fresh oxygen rushes into the lungs once again as the ribcage expands fully. The whale rests for several minutes, replenishing its oxygen reserves before preparing for another descent. From the perspective of physiology, the sperm whale represents one of the most extraordinary solutions to the challenge of pressure ever evolved among mammals. Rather than resisting the crushing force of the ocean, it allows its body to adapt dynamically. The lungs collapse safely, the ribcage folds inward, and oxygen is stored within tissues designed to function even when air is no longer circulating through the respiratory system. This adaptation transforms what would be a deadly environment for most mammals into a hunting ground. The depths that remain inaccessible to other predators become the whale's domain, filled with prey that evolved far from the reach of sunlight. By mastering the physics of pressure, the sperm whale occupies a niche that few other animals can exploit. In the vast vertical landscape of the open ocean, survival often depends on the ability to move between worlds. The sperm whale spends part of its life beneath the bright surface waters where breathing is possible, and part in the crushing darkness thousands of meters below where prey waits in the cold silence. Each dive represents a journey through layers of pressure that would destroy most air-breathing creatures. Yet for the sperm whale, this descent is simply part of its daily routine. The ocean presses inward with enormous force, but the whale's body responds not with resistance but with flexibility. Its lungs collapse safely, its rib cage folds inward like a living pressure tank, and its physiology continues functioning as though the weight of the entire ocean were nothing more than a passing inconvenience. In doing so, the sperm whale demonstrates one of the most remarkable examples of evolutionary engineering found anywhere in the natural world. It is a mammal that carries air-breathing lungs into a realm designed to crush them, surviving depths that would seem impossible through a body capable of transforming under pressure and returning intact each time it rises once again into the light. In the shallow lagoons of tropical oceans, coral reefs rise like living cities from the sea floor, their towers built slowly over centuries by colonies of tiny animals that extract calcium from seawater. Sunlight pours through the clear water above, illuminating a maze of ridges, tunnels, and crevices that shelter thousands of species. Fish flash through open channels like streaks of moving color, while crabs patrol the shadows beneath coral ledges. Yet among this riot of life, one small predator possesses a weapon so powerful that it challenges the limits of biological engineering itself. Hidden inside burrows carved into coral rubble lives the mantis shrimp, an animal whose hunting strike releases forces comparable to those produced by ballistic weapons. At first glance, the mantis shrimp does not appear particularly intimidating. Many species are only the length of a human hand, their bodies segmented like armored insects, with vibrant colors that shimmer beneath the sunlight filtering through the reef. Some display brilliant greens and blues that seem almost electric against the coral background, while others wear muted browns that help them blend into the rubble. Their eyes, mounted on flexible stalks, move independently in constant motion, scanning the environment with an awareness that few predators can match. Yet the true weapon of this animal lies folded beneath its body, hidden within a pair of specialized appendages that resemble compact clubs. These structures are not ordinary limbs. They are biological impact tools designed for extraordinary violence. When a mantis shrimp hunts, it does not slowly pry open shells or patiently tear prey apart. Instead, it unleashes a strike so fast and so forceful that the surrounding water itself momentarily vaporizes from the shock of the blow. The target, often a crab or snail protected by a hard shell, receives the impact with such sudden intensity that the armor shatters instantly. The physics of the strike is astonishing. Within a fraction of a second, powerful muscles within the mantis shrimp's body release energy stored in elastic structures that act like biological springs. These springs accelerate the club forward with tremendous speed. The appendage moves so rapidly that it generates cavitation bubbles in the surrounding water. When those bubbles collapse moments later, they produce secondary shock waves and flashes of light created by the brief release of heat and energy. Even if the initial strike misses, the collapsing cavitation bubble can still damage the prey. For the mantis shrimp, however, delivering such an impact presents an extraordinary challenge. Any structure capable of producing this kind of force must also survive it. If the club were built like the bones of a mammal or the shell of a crab, it would fracture almost immediately under repeated use. Yet the mantis shrimp strikes thousands of times throughout its life without shattering its own weapon. The secret to this durability lies deep within the microscopic architecture of the club itself. When scientists examined the structure of the mantis shrimp's club under powerful microscopes, they discovered a material organization unlike anything previously observed in biology. The internal structure follows a pattern known as a herringbone arrangement, a layered configuration of fibers and mineral crystals that interact in remarkably efficient ways. Instead of forming straight, brittle structures that allow cracks to travel easily through the material, the fibers are arranged in interlocking zigzag patterns that redirect stress throughout the structure. To understand why this matters, it helps to consider how most materials fail. When a crack forms in a rigid material, it tends to spread along predictable lines, often traveling rapidly through the structure until it causes catastrophic failure. In biological systems, this kind of fracture can be disastrous. A broken limb or shell may leave an animal defenseless against predators or unable to feed. The mantis shrimp's club solves this problem through structural complexity at the microscopic level. The herringbone pattern causes stress waves generated by each strike to disperse across the material rather than concentrating in one location. When microscopic cracks begin to form, they encounter layers of fibers oriented in different directions. These layers interrupt the crack's path, forcing it to change direction repeatedly. Each change dissipates energy, preventing the crack from spreading far enough to cause serious damage. This process transforms what might otherwise be a brittle structure into a remarkably resilient one. Instead of shattering under stress, the club absorbs and redistributes the impact forces generated during each strike. The outer surface of the club is also reinforced with a dense mineral layer that resists abrasion and compression, allowing it to withstand repeated collisions with hard shells and coral. The result is a weapon capable of delivering impacts comparable in energy to a small caliber projectile, yet durable enough to survive those forces over and over again. Each strike represents a complex interaction between muscle power, elastic energy storage, and advanced material design, shaped by millions of years of evolutionary refinement. Within the reef ecosystem, this capability gives the mantis shrimp a unique role. Many predators rely on speed or stealth to capture prey, but few possess the mechanical power to break through the hard defenses of shell-bearing animals. Crabs, snails, and clams often depend on their shells as primary protection against attack. Yet when confronted with a mantis shrimp, that protection can become a liability. A mantis shrimp hunting from its burrow watches the surrounding reef with extraordinary visual awareness. Its compound eyes contain a level of complexity rarely matched in the animal kingdom. Each eye can detect subtle differences in color and polarization of light, allowing the shrimp to perceive visual information invisible to most other creatures. This sensory ability helps it detect prey moving across the reef surface or identify rival mantis shrimp approaching its territory. When prey wanders too close, the mantis shrimp emerges suddenly from its burrow. The club unfolds in a movement so fast that it becomes nearly invisible to the human eye. In a fraction of a second, the strike lands with explosive force. The shell of a snail cracks open instantly, or the carapace of a crab splits under the pressure. The mantis shrimp then pulls the stunned prey into its burrow, where it consumes the exposed tissues at its leisure. This strategy allows the mantis shrimp to exploit food sources that remain inaccessible to many other predators. Hard-shelled animals that might resist the claws of fish or the grasp of octopuses become vulnerable targets when confronted with the shrimp's crushing weapon. In doing so, the mantis shrimp influences the structure of reef communities by regulating populations of shelled invertebrates that graze on algae or compete for space within the coral ecosystem. The durability of the mantis shrimp's club also shapes its behavior beyond hunting. These animals are famously territorial, defending their burrows against intruders with remarkable aggression. When rival mantis shrimp encounter one another, they sometimes engage in ritualized combat in which they strike each other's clubs repeatedly in displays of strength. The structural resilience of the club ensures that these contests rarely cause catastrophic injury, even though the forces involved are immense. Scientists studying the mantis shrimp's impact mechanics have been fascinated by the implications for material science. The herringbone structure observed within the club represents a natural solution to a problem engineers have long struggled to solve: how to design materials that resist catastrophic failure under repeated high-stress impacts. By analyzing the way cracks are redirected and energy is dispersed within the mantis shrimp's club, researchers hope to develop stronger composites for use in aerospace engineering, protective equipment, and advanced manufacturing. Yet the mantis shrimp itself remains unaware of the technological inspiration it provides. Within the reef environment, its existence revolves around simple priorities shaped by evolution. It must defend its burrow, capture prey, avoid predators, and reproduce. The extraordinary structure of its weapon is not a deliberate design, but the product of countless generations in which individuals with slightly stronger or more resilient clubs survived long enough to pass their traits forward. As the sun begins to lower over the reef and shadows lengthen across the coral landscape, activity among the animals increases once again. Fish dart between coral branches, and small crustaceans emerge cautiously from hiding places. Within its burrow, the mantis shrimp waits with patient stillness, its eyes rotating independently as they monitor every movement in the surrounding water. The club remains folded beneath its body, a silent promise of the force it can unleash when the moment arrives. Deep within its structure, layers of fibers and minerals arranged in a herringbone pattern prepare to absorb the shock of another strike. Each time the mantis shrimp launches its attack, the forces involved would shatter most biological structures instantly. Yet the club survives, redirecting stress through its intricate architecture and emerging intact, ready to strike again. In the violent and crowded ecosystem of the coral reef, survival often depends on subtle advantages. Some animals rely on camouflage, others on speed, venom, or armor. The mantis shrimp has chosen a different path, becoming a master of impact, whose weapon combines extraordinary power with remarkable resilience. Its clubs represent one of nature's most elegant solutions to the problem of delivering destructive force without destroying the body that produces it. Through the lens of evolutionary design, the mantis shrimp stands as a reminder that even the smallest predators can possess engineering feats, rivaling the most advanced technologies created by human hands. Hidden beneath the coral rubble, armed with a weapon built from microscopic patterns of remarkable sophistication, the mantis shrimp continues its relentless patrol of the reef, striking again and again with a force that few creatures can survive. Along the drifting margins of Arctic sea ice, the world becomes a study in contrasts between movement and stillness. Vast flows of frozen ocean drift slowly across the polar basin, grinding against one another with deep, echoing groans that travel through the ice like distant thunder. Above them stretches a pale sky where the sun, when it appears at all, moves low along the horizon and casts long horizontal light across the frozen landscape. Beneath the ice, however, the ocean remains in constant motion. Dark water circulates slowly through channels and openings between the flows, carrying nutrients and life through an environment that appears lifeless from above. It is within this stark environment that one of the Arctic's most distinctive marine mammals thrives. The walrus rests in heavy groups across the ice, its enormous body draped over the frozen surface like a mound of living stone. Thick folds of skin ripple along its sides as it breathes slowly, occasionally lifting its head to reveal long ivory tusks that curve downward like ancient tools carved from bone. At first glance, the animal appears sluggish and heavy, almost out of place in a world where survival often depends on agility and speed. Yet the walrus is not merely surviving here. It is precisely engineered for one of the most extreme thermal environments on Earth. The Arctic Ocean is a place where water temperatures hover near the freezing point for much of the year. For most mammals, immersion in water this cold would rapidly drain body heat and threaten the stability of internal organs. Water conducts heat away from the body far more efficiently than air, making cold seas particularly dangerous for warm-blooded animals. Yet walruses spend much of their lives swimming and diving through these icy waters in search of food, often remaining submerged for several minutes while exploring the seabed. The key to their survival lies in a remarkable physiological system that allows them to control the flow of blood through their own skin. Unlike many animals whose circulation remains relatively constant, walruses possess an extraordinary network of blood vessels embedded within the thick layers of their skin and blubber. These vessels can expand or contract in response to environmental conditions, allowing the animal to regulate how much heat escapes from its body. At the heart of this system lies a phenomenon known as blood shunting. In cold conditions, the walrus can dramatically reduce the flow of blood to its outer skin layers. When this happens, the blood vessels within the skin constrict, diverting circulation away from the body surface and back toward the animal's core. The effect is immediate and striking. The skin, which normally carries a faint pink coloration from circulating blood, begins to pale as circulation decreases. In extreme cases, the animal's skin can appear almost white, a visual signal that the body is conserving heat with extraordinary efficiency. By limiting blood flow to the skin, the walrus reduces the amount of heat that escapes into the surrounding water. The thick layer of blubber beneath the skin acts as insulation, trapping warmth generated by the animal's metabolism and preventing it from dissipating into the environment. Meanwhile, the blood that continues circulating through the heart, brain, and internal organs remains close to the animal's core temperature, which stays remarkably stable at roughly the same temperature maintained by most mammals. Maintaining this stable internal temperature is critical for the functioning of vital organs. The heart must continue pumping efficiently. The brain must process sensory information and coordinate movement, and the muscles must generate enough power to propel the animal through dense, icy water. If the internal temperature were allowed to drop significantly, these systems would begin to fail. By redirecting blood flow away from the skin, the walrus protects its internal environment even when immersed in water that hovers just above the freezing point. This ability becomes particularly important during long feeding dives. Walruses rely primarily on the rich benthic communities that inhabit the shallow seafloor beneath Arctic waters. Clams, worms, and other invertebrates live buried within sediments, forming dense populations that provide an abundant food source for animals capable of reaching them. To access this food, walruses descend from the surface and search along the seabed, using sensitive whiskers that cover their snouts. These whiskers, known as vibrissae, are among the most specialized sensory structures in the marine mammal world. Each whisker is connected to a complex network of nerves that allow the walrus to detect subtle textures and movements within the sediment. As the animal glides along the seabed, it sweeps its whiskered snout across the surface like a biological sensor array, identifying buried clams hidden beneath layers of sand and mud. Once prey is located, the walrus uses a remarkable feeding technique. It creates powerful suction by sealing its lips around the clam's shell and pulling the soft body from within. The shell itself remains behind on the seabed, while the walrus consumes the nutrient-rich tissue inside. Over the course of a feeding session, a single walrus may consume thousands of clams, leaving behind a patch of disturbed sediment that reveals where the animal has foraged. Throughout this process, the walrus remains submerged in water cold enough to threaten even well-insulated animals. Yet its internal temperature remains stable because the blood shunt system continues to protect the core from heat loss. The skin may grow pale and cool to the touch, but the organs deep within the body remain safely warm. Interestingly, the walrus's ability to regulate blood flow also works in the opposite direction when the animal emerges onto the ice. During periods of rest, large groups of walruses often haul out together on floating ice floes or rocky shorelines. In these gatherings, dozens or even hundreds of massive animals lie pressed closely together, their bodies forming a dense living landscape of wrinkled skin and tusks. Under these conditions, the challenge shifts from conserving heat to releasing it. The thick blubber that protects the walrus in cold water can become a liability when the animal rests in direct sunlight or in close proximity to others. If too much heat accumulates within the body, internal temperature could rise to dangerous levels. To solve this problem, the walrus reverses its circulatory strategy. Blood vessels within the skin expand, increasing the flow of warm blood to the body surface. As this blood circulates through the outer layers of the skin, heat dissipates into the surrounding air. The skin itself becomes flushed with color, sometimes appearing pink or even reddish, as blood fills the vessels near the surface. This rapid shift between constricted and expanded blood flow allows the walrus to function across a wide range of thermal conditions without compromising its internal stability. When submerged in icy water, the body conserves heat by restricting circulation to the skin. When resting on ice or land, it releases heat by increasing blood flow outward. The effectiveness of this system becomes even more impressive when considering the walrus's overall lifestyle. Unlike many marine mammals that rely primarily on speed or agility, walruses often move slowly and deliberately. Their massive bodies, which can weigh more than a small car, require considerable energy to propel through water. Yet this apparent lack of speed is compensated by remarkable physiological resilience. The Arctic ecosystem itself depends heavily on animals capable of enduring such extremes. Beneath the ice, productivity pulses through seasonal cycles of light and darkness. During the brief Arctic summer, sunlight floods the surface waters, triggering blooms of microscopic algae that form the base of the marine food web. These blooms support the growth of countless organisms, from tiny zooplankton to fish, seabirds, and marine mammals. Walruses occupy a unique role within this ecosystem. By feeding heavily on benthic invertebrates, they influence the structure of seafloor communities and recycle nutrients back into the surrounding waters. Their feeding disturbances stir sediments, releasing organic material that can be consumed by other organisms. In this way, the walrus functions not only as a predator, but also as an ecological engineer shaping the dynamics of the Arctic seabed. Predators capable of challenging a full-grown walrus are rare. Polar bears occasionally attempt to attack young or weakened individuals, particularly when the animals rest on ice floes near the edge of open water. Yet the size, tusks, and social behavior of walruses make them formidable opponents. Adults often gather in groups, and their combined presence can deter many predators from approaching. Despite these advantages, survival in the Arctic remains a constant negotiation with the environment. Ice conditions shift with changing seasons, storms break apart flows, and the availability of feeding grounds can fluctuate dramatically. The walrus's physiological adaptations, including its extraordinary blood shunt system, allow it to endure these challenges without losing the delicate thermal balance required by its internal organs. As evening descends over the polar sea and the faint glow of twilight spreads across the horizon, a group of walruses slides slowly from an ice floe into the dark water below. Their massive bodies disappear beneath the surface one by one, leaving behind only ripples that quickly fade into the surrounding stillness. Beneath the water, the cold presses in from all sides. yet their bodies respond instantly by redirecting circulation, preserving warmth where it matters most. In this way, the walrus carries its own climate control system wherever it travels. By manipulating the flow of blood through its skin, it transforms a body vulnerable to cold into one capable of enduring some of the harshest waters on Earth. The skin may pale as circulation retreats inward, but deep within the animal's core, warmth persists steadily, allowing life to continue even in an ocean that hovers on the edge of ice. In the warm coastal waters of tropical seas, where mangrove roots stretch like tangled fingers into the tide and coral reefs form labyrinths of stone and living tissue, the ocean often appears calm and inviting. Sunlight penetrates easily through the clear water, illuminating drifting particles and schools of fish that move like synchronized clouds through the shallows. Yet within this tranquil environment exists a predator that challenges one of the most basic assumptions about life: the idea that intelligence must reside within a brain. Moving silently through these waters is the box jellyfish, a creature that operates with remarkable coordination despite lacking a centralized nervous system. From a distance, the animal appears almost delicate. Its transparent body forms a cube-shaped bell that drifts gently with the movement of the tide, trailing long tentacles that shimmer in the filtered light. Each pulse of the bell propels the animal forward with surprising precision, allowing it to navigate through mangrove channels and coral structures with a level of control that seems unusual for an organism without a brain. Many jellyfish simply drift passively through ocean currents, but the box jellyfish actively steers through its environment, adjusting direction and speed in response to obstacles, prey, and changing light conditions. This ability begins with the structure of the animal itself. Unlike many simpler jellyfish, box jellyfish possess a sophisticated arrangement of sensory organs positioned around the base of their bell. At each corner of the cube-shaped body sits a specialized structure known as a ropalium, a sensory hub containing a remarkable collection of organs capable of detecting light, orientation, and movement. Each ropalium contains multiple eyes, including complex lens-based eyes that resemble miniature versions of the eyes found in more advanced animals. These eyes allow the box jellyfish to perceive contrasts in light and shadow, detect vertical structures such as mangrove roots, and navigate through complex coastal habitats. In shallow tropical lagoons, where roots, coral branches, and seagrass blades form dense underwater forests, such visual awareness is essential. A jellyfish that drifts blindly through these environments would quickly become entangled or damaged. Instead, the box jellyfish glides with subtle control, steering away from obstacles while pursuing small fish and crustaceans that form its primary prey. Yet what makes this sensory system particularly fascinating is not simply the presence of eyes, but the way information from those eyes is processed. In animals with centralized nervous systems, sensory signals typically travel to a brain where they are integrated and interpreted before generating a response. The box jellyfish, however, lacks any structure that resembles a brain in the traditional sense. Instead, its nervous system is arranged as a network distributed throughout the body. Each ropalium functions as a localized processing center. Within these hubs, sensory information from the eyes and other receptors is analyzed directly, allowing the animal to respond rapidly to environmental changes. If one ropalium detects an approaching obstacle, it can trigger adjustments in the contractions of the bell that alter the animal's swimming direction. If another senses a concentration of prey, it can coordinate tentacle movements that increase the chances of capture. This arrangement creates a form of distributed intelligence in which decision-making occurs across multiple locations simultaneously. Rather than relying on a single central command center, the box jellyfish operates through several independent yet interconnected nodes. Each sensory hub acts like a miniature brain, capable of processing information and generating behavioral responses on its own. The advantages of this design become particularly evident when considering the hazards of the animal's environment. The tentacles of the box jellyfish are lined with millions of microscopic stinging cells known as nematocysts, each capable of firing a tiny harpoon tipped with venom. These cells allow the jellyfish to capture prey and defend itself against predators. However, Encounters with larger animals can still cause physical damage. Fish may bite off sections of tentacles, currents may tear delicate tissues, and collisions with debris can injure parts of the bell. In an organism dependent on a centralized brain, such damage could be catastrophic. If the brain were destroyed or severely injured, the entire body would lose coordination and cease functioning. The distributed nervous system of the box jellyfish offers a different form of resilience. Because processing centers exist in multiple locations around the bell, the loss of one region does not necessarily disable the entire animal. The remaining Ropalia continue receiving sensory information and coordinating movement. In essence, the box jellyfish spreads its intelligence across its body. Each corner of the bell contributes to the overall functioning of the organism, creating redundancy that enhances survival. Even when parts of the animal are damaged, the remaining sensory hubs can continue guiding movement and feeding behavior. This decentralized architecture also influences how the jellyfish hunts. When small fish swim near the drifting tentacles, contact with the stinging cells triggers an immediate response. Nematocysts fire almost instantaneously, injecting potent venom that disrupts the prey's nervous system. Immobilized fish become entangled in the tentacles as the jellyfish's bell pulses rhythmically, drawing the prey toward the mouth located at the center of the bell's underside. The venom itself ranks among the most powerful produced by marine animals. It acts rapidly on the nervous and cardiovascular systems of small prey, ensuring that captured fish cannot escape once the stinging cells have fired. For humans who accidentally encounter certain species of box jellyfish, the venom can cause severe physiological reactions. Yet within the ecosystem, this potency serves a clear purpose. It allows the jellyfish to capture fast-moving prey despite lacking jaws, claws, or muscular limbs. The distributed nervous system coordinates these complex interactions without centralized control. Signals generated by tentacle contact travel through nerve networks embedded within the jellyfish's tissues, triggering contractions that draw prey inward while maintaining swimming movements that stabilize the animal in the water column. The Ropalia continue monitoring environmental cues, ensuring that the jellyfish avoids obstacles and remains within productive hunting areas. The box jellyfish's reliance on decentralized processing also shapes its movement patterns within coastal ecosystems. Many species exhibit daily vertical migrations, moving into shallow waters during certain times of day, while retreating to deeper areas during others. These movements appear to be guided by changes in light intensity, detected by the eyes within the Ropalia. As sunlight fades or intensifies, the jellyfish adjust their swimming direction accordingly. In mangrove environments, where roots form dense vertical structures, the jellyfish's visual system becomes particularly important. Studies have shown that box jellyfish actively steer away from dark vertical objects that resemble roots or obstacles. This ability prevents collisions that could damage the delicate bell or entangle the tentacles. The Ropalia detect contrasts in light and shadow, processing this information locally to guide the animal's movements. Even the animal's orientation within the water column is controlled through decentralized sensing. Gravity-sensitive structures within the Ropalia detect changes in the jellyfish's position, ensuring that the bell maintains the correct orientation for effective swimming. This allows the jellyfish to remain upright even when currents attempt to push it off balance. Such coordination might appear to require a centralized brain, yet the box jellyfish accomplishes it through the interaction of multiple small processing centers working simultaneously. Each Ropalium contributes a piece of the puzzle, integrating sensory input and triggering motor responses within nearby tissues. Together, they create a network capable of guiding behavior with remarkable precision. The evolutionary origins of this system reveal much about how nervous systems develop across the animal kingdom. The ancestors of modern jellyfish appeared long before animals evolved complex brains or spinal cords. Early nervous systems likely consisted of simple nerve nets distributed across the body, allowing basic coordination of movement and sensory responses. In the box jellyfish lineage, this ancestral arrangement did not disappear. Instead, it became more specialized, producing localized hubs capable of more sophisticated processing. The result is an organism that demonstrates how intelligence can emerge without centralized control. Rather than building a large brain to manage every function, The box jellyfish relies on cooperation among smaller units spread across its body. This distributed architecture allows the animal to continue functioning even when parts of it are damaged, providing resilience in an environment where injury is common. As the tide moves gently through the mangrove channels and the last light of evening fades beneath the canopy of roots, a box jellyfish glides silently through the dim water. Its tentacles trail behind like threads of glass, nearly invisible against the darkening background. At each corner of its bell, sensory hubs scan the environment, detecting subtle changes in light and motion. The animal pulses forward with steady rhythm, navigating around roots and coral fragments as it searches for prey. There is no brain directing these movements in the way humans might imagine. Instead, multiple centers of perception and control operate simultaneously, each contributing to the animal's survival. In the quiet waters of tropical coasts, the box jellyfish demonstrates that intelligence does not always require a single command center. By distributing awareness throughout its body, it has evolved a form of resilience that allows it to continue hunting, navigating and surviving, even when parts of the organism are lost. In this way, the box jellyfish challenges conventional ideas about the relationship between brains and behavior, revealing that nature often solves complex problems through architectures far different from those found in familiar animals. Across the warm tropical oceans of the Indo-Pacific, where coral reefs stretch along coastlines and open water shimmers beneath the relentless sun, the sea surface often appears calm and endlessly blue. Beneath that surface, however, life unfolds within a chemical environment that presents one of the most persistent challenges any vertebrate must face. Seawater contains a dense concentration of dissolved salt, far more than the internal fluids of most animals can tolerate. For creatures living permanently in this environment, survival requires constant strategies to maintain the delicate balance between water and salt within the body. Among the animals that have solved this challenge in the most unusual ways are the sea snakes. Slender and elegant, their bodies glide effortlessly through the tropical seas as they search for prey among coral reefs, seagrass beds, and the open ocean. Their flattened paddle-shaped tails propel them forward with graceful movements that resemble the strokes of a ribbon drifting through water. Yet despite spending nearly their entire lives immersed in seawater, these reptiles cannot simply drink the ocean around them. Like their terrestrial relatives, sea snakes evolved from ancestors that lived on land. Their bodies still follow the basic physiological rules that govern most reptiles. The fluids circulating within their tissues contain much lower concentrations of salt than the surrounding ocean. If a sea snake were to drink seawater directly, the salt load would overwhelm its internal balance, forcing the animal to expend enormous amounts of energy attempting to remove the excess salt. Over time, dehydration and organ stress would become unavoidable. Instead, sea snakes have evolved two remarkable strategies that allow them to live in salt water without relying on it as a source of drinking water. One strategy involves patience and an intimate awareness of weather patterns in the tropical seas. The other depends on specialized organs capable of removing salt directly from the bloodstream. To understand the first strategy, it is necessary to consider how rain behaves when it falls upon the ocean surface. Tropical storms often sweep across the warm seas where sea snakes live, releasing sudden downpours that can last from minutes to hours. Although rainwater quickly mixes with seawater under most conditions, there is a brief moment when something unusual occurs. Fresh rainwater is less dense than seawater because it lacks dissolved salt. When a heavy rainfall strikes calm ocean water, the fresh water does not immediately mix into the surrounding sea. Instead, it forms a thin floating layer on the surface, sometimes only a few centimeters deep, but large enough to alter the chemistry of the uppermost water. This layer is known as a freshwater lens. For sea snakes, these lenses represent rare opportunities. After extended periods without rainfall, many sea snakes become physiologically dehydrated despite being surrounded by water. Their bodies gradually lose fresh water through normal metabolic processes and through diffusion across their skin. Because they cannot drink the surrounding seawater, they must wait for rain to restore their internal balance. When a tropical storm passes overhead, sea snakes often rise to the surface. Observers have documented these reptiles actively drinking from the freshwater lenses that form during rainfall. The snakes extend their heads above the water, and swallow the rain-diluted surface layer before it has time to disperse. In these moments, the ocean briefly becomes drinkable. The behavior reveals how closely these animals are tied to the rhythms of the tropical climate. In regions where seasonal rains are predictable, sea snakes may rely heavily on these storms to replenish their water supply. The timing of rainfall can influence their movements, feeding patterns, and even reproductive behavior. Yet rain alone cannot solve the entire challenge of salt balance. Even if sea snakes avoid drinking seawater, they still absorb small amounts of salt through the food they consume and through passive diffusion across their skin. Fish and other marine prey contain salt concentrations similar to seawater, and digestion inevitably introduces sodium and chloride ions into the bloodstream. To handle this constant influx of salt, sea snakes possess specialized glands located beneath their tongues. These structures are known as sublingual salt glands, and they function as powerful biological pumps capable of extracting excess salt from the blood. Within these glands, specialized cells actively transport sodium and chloride ions from the bloodstream into a concentrated fluid. This fluid accumulates within small ducts that lead to openings beneath the tongue. When the sea snake extends its tongue or opens its mouth slightly, droplets of this concentrated saline solution are expelled into the surrounding water. The process occurs continuously, maintaining the delicate chemical balance required for normal cellular function. By actively removing salt from the blood, the sublingual glands allow sea snakes to consume marine prey without suffering the toxic consequences of salt accumulation. The efficiency of this system is remarkable. The glands can produce secretions far more concentrated than seawater itself, allowing the snake to expel excess salt with minimal loss of fresh water. In effect, the gland operates as a miniature desalination plant embedded within the animal's head. This combination of rainwater drinking and active salt removal enables sea snakes to thrive in environments where most reptiles would quickly perish. Their bodies maintain internal fluid concentrations similar to those of terrestrial snakes, even while spending nearly their entire lives submerged in salt water. The ability to regulate salt balance also shapes the sea snake's ecological role within tropical marine ecosystems. Many species specialize in hunting small fish that inhabit coral reefs and seagrass beds. These fish often rely on narrow crevices or dense vegetation for protection, yet the slender bodies of sea snakes allow them to pursue prey into spaces inaccessible to larger predators. As the snake glides through the reef, its forked tongue flicks repeatedly through the water, sampling chemical cues left behind by passing fish. These chemical traces guide the snake toward potential prey with remarkable precision. When the target is located, the snake strikes rapidly, injecting venom that immobilizes the fish within seconds. Unlike many terrestrial snakes, sea snakes often swallow their prey underwater. Their lungs extend along much of the body length, allowing them to store oxygen efficiently during dives that may last several minutes. Some species can absorb small amounts of oxygen directly through their skin, further extending their underwater endurance. Despite these impressive adaptations, sea snakes remain closely tied to the surface environment. They must periodically rise to breathe air, and their reliance on freshwater lenses during rainfall creates a subtle dependency on weather patterns. In regions where prolonged dry periods occur, dehydration can become a serious threat. Studies have revealed that during extended drought conditions, sea snakes may show signs of physiological stress. Their blood becomes more concentrated, and their activity levels may decline as they attempt to conserve water. When rain finally arrives, the snakes quickly take advantage of the brief window when freshwater lenses form across the ocean surface. This delicate balance between salt and water reveals how even highly specialized marine reptiles remain connected to processes occurring in the atmosphere above. The tropical storms that sweep across the ocean do more than shape waves and currents. For sea snakes, they provide the fresh water necessary to sustain life. The presence of sublingual salt glands also highlights the remarkable versatility of evolutionary solutions. Rather than abandoning their reptilian physiology entirely, sea snakes adapted existing biological systems to meet the demands of a marine environment. The glands evolved from ancestral structures that once helped terrestrial snakes manage minor fluctuations in salt balance. Over time, those structures became powerful pumps, capable of removing salt far more efficiently than before. As evening settles over the tropical sea and the surface waters grow calm beneath a fading sky, a sea snake drifts upward through the warm blue water. Its slender body undulates gently as it approaches the surface, where faint ripples reflect the last light of the sun. The snake's head breaks through the water for a brief moment as it inhales fresh air before slipping back beneath the surface. Within its body, the quiet work of salt regulation continues. Blood circulates through the sublingual glands, where specialized cells draw out excess sodium and chloride ions. Tiny droplets of concentrated brine are released beneath the tongue and dispersed into the surrounding ocean. Above the water, clouds gather along the distant horizon. In the tropics, weather can change rapidly, and distant storms often arrive with sudden intensity. When rain begins to fall, each droplet will create small ripples across the ocean surface. For a brief time, those droplets will accumulate into a thin lens of fresh water floating atop the salt sea. Somewhere below, sea snakes will sense the change. They will rise toward the surface, tongues tasting the altered chemistry of the water above. In that fleeting layer of rain-diluted ocean, they will drink deeply before the fresh water disappears once again into the vast salt ocean that surrounds them. In this way, the sea snake lives at the intersection of two worlds: the endless salt sea that provides its habitat, and the fleeting freshwater rain that sustains its internal balance. Through patience, physiology, and remarkable evolutionary adaptation, it survives in a place where drinking the water all around it would otherwise be impossible. The seagrass meadow sways slowly in the quiet shallows, each blade bending in rhythm with the tide like a vast underwater field, breathing with the ocean itself. Sunlight spills down through warm tropical water in shifting ribbons of gold and turquoise, illuminating clouds of plankton and the darting movements of tiny fish that weave through the grass like sparks in a breeze. At first glance, nothing here seems built for endurance. Everything appears delicate, fragile, and soft. Yet hidden among these waving stems is a creature whose body contains one of the most remarkable structural secrets in the animal kingdom. Clinging to a blade of seagrass is a seahorse, its small body upright in a posture that would look absurd in almost any other fish. Most fish slice through water horizontally, streamlined and symmetrical, their tails acting as rudders or propellers. The seahorse does the opposite. Its head angles forward like that of a grazing horse, its body armored with bony plates rather than scales, and its tail curls tightly around vegetation like a living hook. It drifts rather than swims, relying on tiny fluttering fins to adjust its position while currents move it gently through its environment. At first glance, the tail appears simple, even fragile, but that impression is profoundly misleading. The tail of a seahorse is not merely a gripping tool. It is a masterpiece of structural engineering, a design so unusual that scientists studying mechanical resilience have turned to it for inspiration when designing flexible robotics and crush-resistant materials. The secret lies in its shape. Most vertebrate tails are cylindrical. Round bones form a column surrounded by muscle and tissue, allowing bending but offering limited protection against crushing forces. When compressed, a round structure distributes pressure evenly around its circumference, but it also leaves little room for controlled deformation. Under enough pressure, such structures fail catastrophically. Bones snap, tissues rupture, and the system collapses. The seahorse tail follows an entirely different blueprint. Instead of being round, its cross-section forms a square. This seemingly simple geometric difference transforms the way the tail behaves under stress. Each segment of the tail is built from interlocking bony plates arranged in a rigid square frame, with flexible joints connecting them like hinges in a mechanical chain. When external pressure is applied, the structure does not collapse inward in a brittle fracture. Instead, the plates slide slightly against one another, absorbing the force and redistributing it along the length of the tail. This stability allows the tail to compress dramatically without breaking. Experiments conducted on preserved seahorse tails have shown that the structure can be squeezed to nearly half its original width while still protecting the internal spinal column. Rather than shattering under pressure, the plates shift, flex, and resist deformation in a controlled way. It is a structural strategy rarely seen in vertebrates, more reminiscent of armored engineering systems than biological anatomy. Understanding why such a structure evolved requires stepping back and examining the ecological life of the seahorse. Unlike most fish, seahorses do not rely on speed or strength to survive. Their bodies are slow, awkward swimmers, incapable of the rapid bursts of motion that allow other species to escape predators or chase prey. Instead, they depend on camouflage, patience, and anchoring themselves securely within complex habitats. Seagrass beds, mangrove roots, coral rubble, and floating algae form the primary landscapes where seahorses live. These environments are dynamic, constantly shifting under the influence of tides, storms, and currents. A fish drifting freely through such waters risks being swept away from feeding grounds or exposed to predators. The seahorse solves this problem by gripping its surroundings. The tail wraps around stems, branches, or strands of vegetation, forming a living anchor. Muscles tighten along the square segments, locking the grip in place with surprising strength. Even strong currents struggle to dislodge a seahorse once it has secured its hold. For an animal that feeds on tiny crustaceans drifting through the water, staying in one location is crucial. The seahorse waits patiently as prey drifts within reach, then snaps its head forward in a rapid suction strike that pulls the victim into its narrow snout. Yet gripping vegetation comes with risks. Coastal habitats experience powerful forces during storms or heavy wave action. Branches whip back and forth, debris collides with anchored animals, and the structures seahorses cling to may twist violently under pressure. In such conditions, a fragile cylindrical tail would easily snap. The square geometry of the seahorse tail, however, transforms these chaotic forces into manageable stress. When a branch or seagrass blade is pushed against the tail, the square plates distribute pressure along multiple edges rather than concentrating it on a single point. The interlocking structure allows slight shifts between segments, preventing sudden fractures. The spine running through the center remains protected by the surrounding armor, ensuring that even severe compression does not damage the nervous system. This resilience becomes especially important in habitats where predators lurk among the vegetation. Many fish species feed on seahorses, including groupers, snappers, and larger wrasses that patrol reef edges and mangrove channels. When threatened, the seahorse does not flee in a burst of speed. Instead, it tightens its grip, twisting its body to align with surrounding plants and relying on camouflage to remain unnoticed. If a predator attempts to pull it loose, the tail functions as both anchor and shield. The square structure resists crushing bites and twisting forces, allowing the animal to remain attached even as it struggles against capture. In some cases, predators may succeed in tearing vegetation away without dislodging the seahorse itself, leaving the animal still gripping a broken fragment as it drifts to a new location. Beyond mechanical resilience, the square tail geometry also enhances the seahorse's remarkable dexterity. Each segment acts like a joint in a flexible robotic arm, allowing the tail to coil, twist, and reposition with fine control. The animal can grip narrow stems, wrap around irregular coral branches, or hold onto floating algae drifting across the open sea. This versatility allows seahorses to exploit a wide range of habitats where stability is essential for feeding and survival. From a biological perspective, the development of this structure represents a fascinating evolutionary compromise. The seahorse lineage belongs to a group of fishes known as syngnathids, which also includes pipefish and sea dragons. Many of these relatives display elongated bodies and specialized snouts designed for suction feeding. However, not all of them possess the square armored tail seen in seahorses. Comparative anatomical studies suggest that as seahorses adopted an upright posture and sedentary lifestyle, natural selection favored individuals capable of gripping vegetation more effectively. Over countless generations, the bones surrounding the spinal column gradually shifted shape, forming protective plates arranged in square configurations. Muscles and connective tissues adapted to this framework, transforming the tail into a flexible yet durable gripping tool. The result is a structure that combines rigidity and flexibility in an elegant balance. Too much rigidity would prevent the tail from coiling around narrow objects. Too much flexibility would leave the spinal column vulnerable to crushing forces. The square-segmented architecture solves this dilemma by allowing controlled deformation under pressure while maintaining overall structural integrity. The implications of this design extend beyond the survival of a single species. Engineers studying biological materials have examined seahorse tails as models for constructing crush-resistant devices. Flexible robotic arms, underwater exploration tools, and even protective gear have drawn inspiration from the interlocking square segments that allow the tail to bend without breaking. In this way, the quiet resilience of a small fish hidden among seagrass may influence technologies used far beyond the ocean. Meanwhile, the seahorse continues its patient existence in the shifting shallows. Anchored to a strand of vegetation, it waits as currents deliver drifting prey. Tiny shrimp glide past in bursts of transparent motion, and the seahorse's head snaps forward with astonishing speed, creating a sudden vacuum that pulls the prey into its narrow mouth. The strike lasts less than the blink of an eye, a brief moment of explosive movement in an otherwise tranquil animal. Around it, the ecosystem pulses with life. Juvenile fish seek refuge among the seagrass blades. Crabs crawl along the sediment below, scavenging fragments of organic debris. In the distance, the silhouettes of larger predators patrol the outer edge of the meadow, where open water meets the sheltering vegetation. The seahorse remains still, its body blending perfectly with the color and texture of the plants it grips. Storms will come, as they always do in coastal waters. Waves will crash against the shore, currents will surge through the meadow, and debris will twist violently through the tangled vegetation. Yet when these forces arrive, the seahorse will not attempt to escape. Instead, it will do what its body has evolved to do with remarkable efficiency. It will tighten its grip, curling its square tail around a stem of seagrass, trusting in a structural design that allows it to endure pressures that would shatter more fragile forms. In that moment, the animal becomes less like a fish and more like a living mechanism embedded within the ecosystem. Its body absorbs stress, redistributes force, and protects the delicate nervous system running through its center. The square tail does not merely hold the seahorse in place. It transforms the animal into a resilient anchor in a constantly moving world, an example of how evolution can reshape anatomy into forms that appear improbable, yet function with extraordinary precision. Amid the shifting water and swaying grass, the seahorse continues its quiet watch, a creature whose survival depends not on speed or aggression, but on geometry. In the architecture of its tail lies a design capable of resisting forces that would destroy simpler structures, a reminder that even in the delicate shallows of the ocean, resilience often hides within the smallest and most unexpected forms. In the labyrinth of a coral reef, where sunlight fractures into thousands of shifting beams and every crevice hides an ecosystem of its own, survival often depends less on speed and more on tolerance. The reef is not merely a garden of color and life. It is also a dense microbial battleground. Bacteria coat nearly every surface. Fungi drift through the water column. Parasites seek entry into wounds and gill tissue. Organic matter accumulates in pockets where currents cannot easily flush it away, creating zones rich with microscopic life. For most animals, living deep within such environments requires constant vigilance against infection and disease. Among the twisting branches of coral and the jagged architecture of limestone skeletons, a long, muscular body slides silently through the narrowest cracks in the reef. Its head appears first, emerging slowly from a shadowed hole between coral heads. The animal's mouth opens and closes rhythmically, revealing rows of needle-like teeth. To a casual observer, this movement may look threatening, as though the creature is preparing to strike. In reality, it is simply breathing. Unlike many fish, this predator cannot pump water over its gills without opening its jaws repeatedly, a motion that has earned it an unfair reputation as a perpetually aggressive hunter. The animal is a Moray eel, one of the reef's most enigmatic predators. Its body is long and serpentine, often patterned with mottled browns, greens or yellows that blend perfectly with the reef's complex textures. Its skin lacks the scales that cover most fishes. Instead, It is wrapped in a thick layer of mucus, a slick coating that glistens under shifting sunlight. At first glance, this slime might seem like little more than lubrication, allowing the eel to slide effortlessly through narrow crevices where rigid-bodied predators cannot follow. Yet this slippery layer conceals a biochemical defense system as remarkable as any physical armor. Coral reefs are microbial hotspots. Warm water accelerates bacterial growth, and the abundance of organic material provides endless fuel for microbial communities. Fish that scrape against coral often acquire tiny cuts and abrasions. These wounds, though small, can become gateways for infection. For animals that spend their lives wedged into reef cavities, where water circulation is limited and microorganisms flourish, such infections could quickly become fatal. Moray eels have solved this problem in an elegant and surprisingly sophisticated way. Their mucus layer contains a complex mixture of antimicrobial compounds known as peptides. These molecules function as natural antibiotics, capable of destroying bacteria and fungi that attempt to colonize the eel's skin. When microorganisms come into contact with the slime, the peptides disrupt their cell membranes, causing them to rupture and die before they can establish an infection. This biochemical shield transforms the eel's entire body surface into a defensive barrier against microbes. Even when the eel scrapes against rough coral or pushes through sediment filled with bacteria, its skin remains protected. Laboratory studies examining Moray mucus have revealed that many of these antimicrobial peptides are unique, meaning they differ from compounds found in other marine animals. Some researchers believe these molecules may even inspire future medical treatments, as they demonstrate powerful antibacterial effects against strains that resist conventional antibiotics. The ecological consequences of this adaptation are profound. Because the eel is largely immune to the microbial hazards of reef crevices, it can occupy habitats that would be hostile to other predators. Many reef fish prefer open water or the outer edges of coral structures where currents continually flush away waste and microorganisms. The Moray eel, by contrast, thrives in the reef's tightest and darkest cavities. These spaces are often avoided by other hunters, creating a network of hidden corridors where the eel can move unseen. From within these narrow passages, the eel becomes an ambush specialist. Its body coils deep inside coral tunnels, leaving only its head protruding into the open water. Small fish, crustaceans, and octopuses pass nearby, often unaware that they are drifting within reach of a predator concealed just centimeters away. When prey ventures too close, the eel lunges forward with startling speed. Yet the Moray eel's hunting method contains another evolutionary innovation that reflects the constraints of its body design. Most fish capture prey by creating suction, rapidly expanding their mouths to pull water and prey inward. The elongated skull of the Moray eel limits this ability. Instead, the eel relies on a second set of jaws hidden deep within its throat. These specialized structures, known as pharyngeal jaws, move forward after the initial bite, gripping the prey and pulling it deeper into the mouth. This mechanism resembles a biological conveyor system. The outer jaws seize the prey first. Then the pharyngeal jaws slide forward, clamp down and retract, dragging the victim into the eel's throat. The process repeats until the prey disappears entirely into the digestive tract. It is a feeding strategy that compensates for the eel's inability to generate strong suction, allowing it to consume prey even within the confined spaces of coral tunnels. As the eel retreats back into its crevice after a successful hunt, its mucus layer continues to perform its quiet protective role. Tiny parasites drifting through the water attempt to attach themselves to the eel's skin, but the antimicrobial compounds disrupt their survival. Bacteria that might exploit microscopic abrasions are neutralized almost instantly. In an environment where infection can spread rapidly through stressed or injured animals, this chemical shield grants the eel remarkable resilience. The relationship between Moray eels and the microbial world around them also highlights an important principle of reef ecology. Coral reefs are often celebrated for their visible diversity of fish, corals, and invertebrates. But beneath that colorful surface lies an even more complex universe of microscopic organisms. Bacteria, algae, and fungi form intricate networks that influence nutrient cycles, coral health, and the survival of countless species. For predators like the Moray eel, navigating this microbial landscape requires not only physical strength, but biochemical defenses that operate continuously at the cellular level. Interestingly, the eel's slime layer also affects its interactions with other reef inhabitants. Cleaner fish and cleaner shrimp, small animals that feed on parasites and dead tissue from larger hosts, often approach Moray eels with remarkable confidence. These cleaners enter the eel's open mouth or glide along its body, removing debris and parasites that the mucus layer has already weakened or immobilized. The eel remains surprisingly tolerant of these visitors, allowing them to complete their work without aggression. This partnership benefits both species. The cleaners receive a steady food source, while the eel gains additional maintenance for areas of its body that might otherwise accumulate parasites. The antimicrobial mucus does not eliminate the need for such cleaning interactions, but it reduces the risk of infection during the process, ensuring that minor abrasions or parasite removal do not become entry points for harmful microbes. Beyond the reef's daylight hours, the Moray eel's resilience becomes even more evident. As dusk settles and shadows stretch across the coral landscape, many reef predators emerge to hunt. The eel glides through narrow passages between coral heads, its body weaving through openings barely wider than its own width. Each movement brings it into contact with rough limestone surfaces and sediments filled with bacteria. Yet the eel's protective mucus layer ensures that these constant abrasions rarely lead to infection. This ability to withstand the reef's microbial hazards allows Moray eels to live long lives within stable territories. Some individuals remain within the same reef structure for years, memorizing the maze of tunnels and cavities that define their home range. Within these hidden corridors, they become dominant predators, capable of ambushing prey that attempts to hide from faster hunters in open water. Even interactions with larger predators illustrate the value of the eel's biological defenses. Sharks and large groupers occasionally attempt to capture Moray eels, but the eel's slippery body and narrow refuge spaces make such attacks difficult. If the eel manages to wedge itself deep within a coral crevice, predators often abandon the pursuit rather than risk injuring themselves against sharp coral edges. During such struggles, the eel's mucus again plays a crucial role. The slime not only reduces friction, but also discourages microbial colonization of any minor wounds that might occur during the encounter. In a reef environment where damaged tissue can quickly attract harmful bacteria, this chemical protection significantly increases the eel's chances of recovery. From a scientific perspective, the study of Moray eel mucus has revealed insights that extend far beyond marine biology. The antimicrobial peptides found within the slime demonstrate powerful activity against a wide range of pathogens, including bacteria that have developed resistance to traditional antibiotics. Researchers exploring these compounds hope they may one day contribute to new treatments for infections that challenge modern medicine. This possibility underscores a broader truth about coral reef ecosystems. Many of the biochemical innovations that protect reef animals from disease may also hold potential benefits for human health. The ocean's evolutionary laboratory has produced molecules with extraordinary properties, shaped by millions of years of competition between microbes and the organisms that resist them. Yet, within the reef itself, these molecular defenses serve a far simpler purpose. They allow a predator with an elongated, vulnerable body to thrive in an environment filled with microbial threats. The Moray eel does not possess the thick armor of a crustacean or the rapid escape speed of many reef fish. Instead, its survival depends on a subtle but powerful chemical barrier that coats every centimeter of its skin. As the tide shifts and currents carry fresh plankton through the reef, the eel returns once again to its familiar crevice. Its head protrudes from the opening, jaws opening and closing rhythmically as water flows across its gills. Small fish pass cautiously nearby, aware of the danger hidden within the shadows, but unable to predict the precise moment when the eel might strike. Around it, the reef continues its endless cycle of growth, decay, and renewal. Coral polyps extend their tiny tentacles into the current. Algae grow across sunlit surfaces. Bacteria multiply in the warm water, forming invisible clouds that drift through every crevice. Within this microbial sea, the Moray eel remains protected by the silent chemistry of its own body. The mucus coating its skin is more than a simple lubricant. It is a living shield, a biochemical defense forged through countless generations of evolution. In a world where infection can spread as quickly as any predator's bite, the eel carries its protection with it at all times, transforming the surface of its body into an invisible barrier against the unseen dangers of the reef. Here, among coral towers and shadowed tunnels, resilience does not always come from strength or speed. Sometimes it comes from chemistry, from molecules so small they cannot be seen with the naked eye, yet powerful enough to protect a predator that spends its life moving through one of the most microbially dense environments on Earth. Far from the shelter of coastlines, the open ocean stretches across immense distances where water and sky merge into a horizon that appears endless. Beneath the rolling surface lies a vertical world of drifting life, currents that move like invisible rivers, and migrations that unfold across thousands of kilometers each year. In this immense blue expanse, the largest animal ever known to exist moves with quiet authority. The blue whale glides through the water with slow, deliberate strokes of its immense tail, its body longer than many buildings, and heavier than any creature that has walked the earth. At first glance, the scale of the animal seems almost impossible. Its heart alone is the size of a small vehicle, beating steadily as it propels blood through arteries thick as a human arm. Each mouthful of seawater contains swarms of tiny crustaceans known as krill, organisms barely the length of a fingernail that gather in dense clouds across the ocean's productive regions. To sustain a body of such immense proportions, the blue whale must consume staggering quantities of these tiny creatures, filtering them through plates of baleen that hang from its upper jaw like enormous curtains. The whale's daily routine is a rhythm shaped by the seasonal productivity of the ocean. In polar waters during the summer months, sunlight floods the sea surface for long hours each day. This burst of energy fuels blooms of microscopic algae that drift through the upper layers of the water column. Krill gather to feed on these blooms, forming dense swarms that can stretch for kilometers beneath the surface. The blue whale arrives in these regions precisely when this abundance reaches its peak. When the whale locates a swarm, it accelerates upward through the cloud of prey with its mouth open wide. The throat expands dramatically as folds of elastic skin along the underside of the body stretch outward, allowing the whale to engulf enormous volumes of water. Once the mouth closes, the whale forces the water outward through the baleen plates using its tongue, trapping thousands of krill inside. The prey is then swallowed in a concentrated mass that provides the energy required to sustain the animal's immense metabolism. The sheer scale of the blue whale raises profound questions about biology. Every organism is built from cells, the microscopic units that carry the genetic instructions for life. In humans and most other animals, these cells divide repeatedly throughout life in order to repair tissues and replace worn structures. Yet each time a cell divides, there exists a small possibility that errors will occur in the replication of DNA. Over time, the accumulation of such errors can lead to uncontrolled cell growth, a condition known as cancer. In smaller animals with relatively few cells, the probability of such errors occurring somewhere within the body remains limited. But the blue whale presents a fascinating paradox. Its body contains vastly more cells than a human body, an almost unimaginable number of individual units constantly maintaining the tissues of this enormous organism. From a simple statistical perspective, one might expect the probability of cancer to increase dramatically with body size. More cells mean more opportunities for mutations to occur. Yet the blue whale does not appear to suffer from cancer at rates higher than those seen in much smaller animals. In fact, observations suggest that these giants experience surprisingly low levels of cancer despite their immense size and long lifespans. This puzzling phenomenon has become known among scientists as Peto's paradox, named after the researcher who first recognized the contradiction between expected cancer rates and the observed health of large animals. The solution to this paradox lies within the whale's genetic defenses. Over millions of years, the evolutionary lineage that produced the blue whale has developed powerful mechanisms to suppress the formation of tumors. Among the most important of these mechanisms are genes responsible for detecting and repairing damaged DNA. In particular, The gene known as P53 plays a crucial role in monitoring the integrity of cellular genetic material. In many animals, this gene acts as a guardian of the genome. When DNA damage occurs within a cell, P53 can trigger a cascade of responses that either repair the damage or initiate a process known as apoptosis, in which the cell effectively self-destructs before it can become dangerous. This mechanism prevents damaged cells from multiplying and forming tumors. The blue whale and its close relatives possess multiple copies of these tumor suppressor genes. Instead of relying on a single version of the gene to monitor cellular health, the whale's genome contains additional layers of protection. Each copy acts as an independent safeguard capable of detecting genetic damage and activating protective responses. The result is a system in which potentially dangerous mutations are more likely to be identified and neutralized before they can spread. This enhanced genetic surveillance dramatically reduces the risk that any one damaged cell will develop into a tumor. In effect, the blue whale's enormous body is protected by a network of molecular watchdogs that constantly patrol its tissues, ensuring that the immense population of cells remains under strict control. The importance of these defenses becomes even clearer when considering the lifespan of these animals. Blue whales can live for many decades, spending years migrating between feeding grounds in polar waters and breeding areas in warmer regions of the ocean. During these long lives, their cells undergo countless cycles of division and renewal. Without strong safeguards, the accumulation of genetic errors over time could pose a serious threat to the stability of the organism, yet the whale's genome has evolved precisely to counter this risk. The additional tumor suppressor genes function as redundant layers of defense, making it far more difficult for cancerous growths to establish themselves within the body. This system allows the whale to maintain cellular stability across a vast population of cells that must continue functioning for decades. The ecological implications of this adaptation extend beyond the health of individual whales. The blue whale occupies a crucial role within marine ecosystems as one of the ocean's largest consumers of krill. By feeding on enormous quantities of these tiny crustaceans, whales influence the distribution and dynamics of plankton communities that form the base of marine food webs. After feeding in polar waters, blue whales release nutrient-rich waste that fertilizes the surrounding ocean. This material contains elements such as iron and nitrogen that stimulate the growth of microscopic algae. These algae, in turn, support the krill populations that whales depend upon. In this way, the whales help sustain the productivity of the ecosystems that feed them, creating a feedback loop that supports the health of entire marine regions. The whale's enormous body also plays a role in transporting nutrients across vast distances. During migrations between feeding and breeding grounds, nutrients consumed in one region are released in another, redistributing essential elements across the ocean basin. This process contributes to the cycling of nutrients in ways that smaller animals cannot replicate. Despite these remarkable adaptations, the blue whale remains vulnerable to threats that originate not from natural processes, but from human activity. In the past, industrial whaling dramatically reduced global populations of these animals. Modern threats include ship strikes, noise pollution, and changes in ocean ecosystems driven by climate shifts that affect krill distribution. Yet the biological resilience of the species remains evident in the mechanisms that protect its cells from disease. The same evolutionary processes that allowed these giants to grow to extraordinary sizes also equipped them with sophisticated defenses against the molecular dangers associated with large bodies and long lifespans. When a blue whale rises toward the surface to breathe, its immense form breaks through the water with quiet power. The exhalation produces a tall column of mist that hangs briefly in the air before drifting away on the wind. Beneath the surface, the whale's tissues continue their silent work. Billions of cells performing the countless tasks required to sustain life within such an enormous body. Each of those cells carries genetic instructions shaped by millions of years of evolution, instructions that include extra safeguards against the chaos that uncontrolled growth can produce. Through the presence of multiple tumor suppressor genes, the whale's body maintains a balance between growth and control that allows it to exist at a scale rarely seen in the history of life. In the immense open ocean, where distances stretch beyond sight and the rhythms of life unfold across seasons and migrations, The blue whale represents one of nature's most extraordinary biological achievements. Its body is not only the largest ever built by evolution, but also one of the most carefully protected at the cellular level. Through the paradox that bears its name, it reminds us that size alone does not determine vulnerability. Instead, evolution can reshape the rules of biology, equipping even the largest creatures with the tools needed to remain healthy in a world where the smallest cellular error could otherwise prove fatal. In the warm shallows of a tropical lagoon, where coral heads rise like ancient fortresses from a pale sandy bottom, life unfolds in a constant negotiation between hunger and survival. Small fish flicker through shafts of sunlight, darting between coral branches, with the urgency of creatures that understand how quickly the calm surface of the reef can erupt into violence. Crabs patrol the rubble fields. Shrimp wave their antennae from the mouths of burrows. Overhead, schools of silvery baitfish move like liquid clouds, changing direction in perfect unison as shadows glide beneath them. Within this intricate web of movement drifts a creature that appears almost comically vulnerable. Its body is rounded and soft, lacking the sleek, hydrodynamic form that allows most reef fish to accelerate away from danger. Its fins paddle gently, propelling it forward with a slow, deliberate motion that seems ill-suited for a place where speed often determines survival. The animal pauses frequently, hovering above patches of coral rubble, inspecting the surface with curious eyes that swivel independently in subtle, careful movements. This is the pufferfish, a species that at first glance seems to embody the opposite of the violent efficiency that dominates much of reef ecology. Yet beneath this harmless appearance lies one of the most potent chemical arsenals found anywhere in the natural world. The pufferfish carries within its tissues a compound so lethal that even the smallest dose can stop a vertebrate nervous system in moments. The chemical is tetrodotoxin, a neurotoxin that interferes with the electrical signals that allow muscles and nerves to function. For most animals, exposure to this compound results in paralysis so complete that breathing itself becomes impossible. Tetrodotoxin does not merely irritate predators or create mild illness. It blocks the very channels in nerve cells that allow sodium ions to flow across cell membranes. Without that flow, electrical impulses cannot propagate along nerves. Muscles receive no signals. The body becomes locked in a silent, immobile state. In many cases, the victim remains conscious even as paralysis spreads through the body, a chilling reminder of how precisely this toxin disrupts biological systems. Yet the most remarkable aspect of this chemical weapon is not its potency. It is the fact that the pufferfish itself remains completely unaffected by the poison saturating its own organs. In many species, the liver, skin, and reproductive tissues contain concentrations of tetrodotoxin strong enough to kill dozens of adult humans. For the fish, however, this lethal compound poses no threat at all. The reason lies deep within the architecture of its cells. Every nerve impulse in an animal's body depends on specialized proteins embedded in cell membranes. These proteins, known as sodium channels, act like microscopic gates that open and close in response to changes in voltage. When the gates open, sodium ions rush into the cell, triggering a cascade of electrical activity that travels along nerves and into muscles. Tetrodotoxin functions by binding tightly to these channels, blocking the passage of sodium ions and halting electrical signaling. In most animals, the toxin fits into the sodium channel like a key in a lock. Once bound, it prevents the channel from opening, effectively shutting down the entire electrical communication system of the nervous system. But in pufferfish, the structure of the sodium channel has been subtly altered by evolution. A tiny mutation in the protein's molecular structure changes the shape of the binding site just enough to prevent tetrodotoxin from attaching securely. This single structural modification transforms the relationship between the fish and the toxin it carries. Where other animals would experience instant paralysis, the pufferfish continues to function normally. Its nerves fire, its muscles contract, and its brain coordinates movement with no interference from the chemical saturating its tissues. The toxin that would destroy most nervous systems becomes, for the pufferfish, an inert passenger within its body. The ecological implications of this mutation are profound. Carrying such a powerful toxin provides the fish with an extraordinary defense against predation. Many reef predators learn quickly that attempting to eat a pufferfish can be a fatal mistake. Even a small bite may introduce enough toxin into the predator's bloodstream to cause paralysis. In some cases, predators that capture pufferfish die within minutes, their nervous systems overwhelmed by the chemical barrier that protects the fish. But the pufferfish does not rely on poison alone. Its defensive strategy combines chemistry with behavior in a remarkable display of biological ingenuity. When threatened, the fish rapidly inhales water into a specialized elastic stomach, inflating its body into a near spherical shape. This sudden transformation increases its apparent size and makes it difficult for predators to swallow. Sharp spines that normally lie flat against the skin extend outward, turning the inflated fish into a prickly ball that few predators can grip effectively. Inflation alone might discourage some attackers, but the real deterrent lies beneath the skin. Any predator that persists long enough to bite through the inflated body risks exposure to tetrodotoxins stored within the fish's tissues. Over time, many predators learn to avoid the distinctive shape and movements of pufferfish altogether. This learned avoidance creates a powerful ecological shield around the species, allowing it to forage openly across reef surfaces where more vulnerable fish would be quickly hunted down. The origin of the toxin itself reveals another fascinating dimension of this evolutionary relationship. Contrary to what one might expect, pufferfish do not produce tetrodotoxin entirely on their own. Instead, the compound is believed to originate from symbiotic bacteria living within the fish's body and diet. These microorganisms synthesize the toxin as part of their metabolic processes, and the fish accumulates it through feeding and internal microbial activity. This partnership illustrates a recurring theme in marine evolution, where chemical defenses often arise through collaborations between animals and microscopic organisms. The bacteria benefit from a stable environment within the host, while the host gains access to a potent chemical weapon that would otherwise be impossible to produce alone. Over countless generations, natural selection has refined this alliance, ensuring that the fish's physiology can tolerate and store the toxin safely. Interestingly, not all pufferfish contain the same levels of tetrodotoxin. Individuals raised in environments without toxin-producing bacteria may develop little or no toxicity at all. In the wild, however, the complex microbial communities of coral reefs and coastal ecosystems provide ample opportunities for these symbiotic relationships to flourish. As a result, many pufferfish become walking reservoirs of one of the most powerful neurotoxins known to science. This chemical defense does more than deter predators. It also shapes the behavior of other organisms within the reef ecosystem. Certain specialized predators have evolved partial resistance to tetrodotoxin, allowing them to consume pufferfish without suffering lethal consequences. Some species of sharks, for example, appear capable of tolerating small doses of the toxin, though even they approach pufferfish cautiously. Meanwhile, other reef inhabitants interact with pufferfish in more subtle ways. Cleaner shrimp and small reef fish sometimes approach puffers to remove parasites from their skin. Despite the presence of toxin within its tissues, the fish's mucus layer and behavior allow these cleaning interactions to occur without harm to either participant. Such partnerships illustrate the delicate balance of cooperation and caution that defines life on a coral reef. As the pufferfish continues its slow patrol across the reef floor, it investigates crevices and coral surfaces with careful precision. Its diet consists primarily of hard-shelled invertebrates such as crabs, mollusks, and sea urchins. To access these prey items, the fish relies on a set of powerful fused teeth that form a beak-like structure capable of crushing shells and exoskeletons. Each bite produces a sharp cracking sound as the fish breaks apart its meal. The ability to consume such tough prey provides an ecological advantage in environments where softer foods may be scarce. Many reef fish compete for plankton or algae, but the pufferfish occupies a niche focused on armored invertebrates that few predators can easily exploit. By crushing these organisms, the fish helps regulate populations of species that might otherwise overgraze coral or dominate reef substrates. Despite its formidable defenses, the pufferfish remains a participant in the complex balance of reef life. Its movements stir sediments, its feeding habits influence invertebrate communities, and its presence signals to predators that certain meals come with dangerous consequences. In this way, the fish contributes to the subtle equilibrium that maintains the diversity of coral reef ecosystems. As the sun begins to sink toward the horizon, the colors of the reef deepen into richer shades of blue and gold. Shadows lengthen across the coral landscape, and nocturnal predators prepare to emerge from their hiding places. The pufferfish drifts toward a sheltered crevice where it will spend the night, its body blending into the mottled textures of coral and rock. Within its tissues, the silent chemistry of tetrodotoxin remains unchanged, a lethal compound stored safely within a body that has evolved the rare ability to ignore its own poison. The mutation in the fish's sodium channels continues to shield its nervous system from the toxin's effects, allowing it to carry a weapon capable of paralyzing creatures many times its size. In the grand theater of the reef, where survival often depends on speed, camouflage, or brute strength, the pufferfish represents a different kind of power. Its dominance comes not from physical force, but from molecular adaptation. By altering the structure of a single protein within its nerve cells, evolution has transformed a deadly toxin into a protective shield, allowing the fish to move through a world of predators with the quiet confidence of an animal that carries its own chemical fortress. Here, among the swirling currents and living architecture of coral, the pufferfish drifts almost peacefully, a slow-moving creature in a fast-paced ecosystem. Yet hidden within its body lies one of nature's most astonishing examples of biochemical resilience, a reminder that sometimes the most unstoppable forces in the ocean are not the largest predators or the fastest hunters, but the creatures that have mastered the invisible language of chemistry. In the dim blue twilight of the continental shelf, where sunlight fades into cold gradients of shadow and the ocean floor begins its long descent toward deeper water, the seafloor appears almost motionless. Fields of sponges rise like weathered sculptures from rocky outcrops. Feather stars extend delicate arms into slow-moving currents. Scattered shells and fragments of coral lie half-buried in fine sediment, evidence of countless lives that have come and gone in this quiet underwater landscape. Yet beneath this stillness, something moves with deliberate precision, its body reshaping itself moment by moment as it glides across the seabed. An octopus emerges slowly from behind a cluster of rocks, its skin shifting color and texture in subtle ripples that mimic the patterns of the surrounding environment. One moment it resembles the mottled surface of the stones beneath it, and the next its skin darkens to match the shadow cast by a nearby ledge. The animal moves without haste, each arm extending forward in fluid motions that appear almost thoughtful, as though the creature is testing the world before committing its weight to the next step. The octopus belongs to a lineage of animals that has followed an evolutionary path dramatically different from that of vertebrates. Its body contains no bones, no rigid skeleton, and no protective shell. Instead, it relies on a muscular hydrostatic system that allows its arms to bend, twist, and compress with extraordinary flexibility. Each arm is lined with rows of suction cups capable of detecting chemical signals in the water, giving the octopus the ability to taste the surfaces it touches. But beneath this remarkable anatomy lies an even more extraordinary biological phenomenon, one that transforms the octopus into one of the most adaptable creatures in the ocean. The waters surrounding this rocky habitat are cold, far colder than the tropical reefs where many marine animals thrive. Temperatures fluctuate with changing currents, sometimes dropping to levels that challenge the basic functioning of nervous systems. In most animals, the electrical signals that travel along nerves slow dramatically as temperatures fall. Chemical reactions proceed more slowly in cold environments, and proteins responsible for transmitting nerve impulses lose efficiency. For creatures whose survival depends on quick responses and precise control of movement, such conditions can be debilitating. Yet the octopus moves with calm confidence through this cold water, its arms exploring the seafloor with delicate coordination. The explanation for this resilience lies deep within the molecular machinery of its nervous system. Unlike most animals, whose genetic instructions remain largely fixed once development is complete, the octopus possesses the ability to modify how its genes are used in real time. This process, known as ribonucleic acid editing, allows the animal to adjust the structure of proteins produced by its cells without altering the underlying genetic blueprint stored in its deoxyribonucleic acid. To understand why this matters, it helps to consider the fundamental relationship between genes and proteins. In most organisms, genes stored within deoxyribonucleic acid provide instructions for building proteins, the molecular machines that carry out nearly every function within a cell. These instructions are copied into ribonucleic acid molecules, which serve as templates guiding the assembly of amino acids into specific protein structures. Once produced, the protein's shape determines how it behaves within the cell. In cold environments, however, the shape and flexibility of proteins become critically important. Many proteins that function well at warm temperatures become sluggish or unstable when the surrounding water cools. For animals with rigid genetic programming, adapting to such changes requires evolutionary timescales, often involving generations of natural selection to refine new protein structures suited to colder conditions. The octopus, remarkably, has evolved a different solution. Its cells possess enzymes capable of editing ribonucleic acid molecules after they are produced. These enzymes alter specific chemical bases within the ribonucleic acid sequence, subtly changing the instructions used to assemble proteins. The resulting proteins differ slightly in their structure from those originally encoded in the animal's genes. This ability allows the octopus to adjust the properties of its proteins in response to environmental conditions. In cold water, certain proteins involved in nerve signaling are modified so that they continue to function efficiently despite the reduced temperature. Ion channels that regulate the flow of charged particles across nerve cell membranes become more flexible, allowing electrical signals to travel along nerves without the delays that cold normally causes. The consequences of this molecular flexibility extend throughout the octopus's nervous system. Unlike most animals, whose brains control the majority of their movements directly, the octopus distributes much of its neural processing throughout its arms. Each arm contains dense networks of neurons capable of coordinating complex movements independently of the central brain. This decentralized system allows the octopus to manipulate objects, explore crevices, and capture prey with extraordinary dexterity. Maintaining the efficiency of such a distributed nervous system in cold water presents significant challenges. Every arm relies on rapid electrical communication between neurons and muscles. If those signals slowed too much, the animal would lose its ability to coordinate movements, leaving it vulnerable to predators and unable to hunt effectively. Through ribonucleic acid editing, the octopus ensures that the proteins responsible for these signals remain optimized for the surrounding temperature. The process unfolds continuously within the animal's cells. Enzymes scan ribonucleic acid molecules, identifying specific sites where chemical alterations will produce beneficial changes in the resulting proteins. These modifications occur after the genetic instructions have been copied, but before the proteins are assembled, allowing the octopus to fine-tune its physiology without altering the permanent record stored in its genes. This dynamic system effectively allows the octopus to adapt its nervous system on the fly. When temperatures drop, the editing enzymes increase their activity at key sites within ribonucleic acid sequences, producing proteins better suited to colder conditions. When temperatures rise again, the pattern of editing shifts accordingly. It is a level of molecular flexibility rarely observed in complex animals. As the octopus continues its slow exploration of the seabed, it pauses near a crevice where the scent of prey drifts faintly through the water. One arm extends cautiously into the narrow opening, its suction cups probing the darkness within. The arm moves with quiet precision, guided by neural circuits capable of making decisions locally without waiting for instructions from the central brain. A moment later, the arm recoils slightly, its suckers gripping the smooth shell of a hidden crab. In an instant, the rest of the animal springs into action. Arms coil around the crevice entrance, anchoring the octopus as it pulls the crab from its hiding place. The prey struggles briefly, but the octopus's beak-like jaws deliver a decisive bite that injects venom capable of immobilizing the crustacean. The entire sequence unfolds with fluid efficiency, demonstrating the remarkable coordination that defines octopus hunting behavior. Such coordination would be impossible if cold water interfered with the speed of neural signaling. Each arm must communicate constantly with neighboring arms and with the central brain. Muscle contractions must occur in precise sequences to maintain grip and balance. By editing the ribonucleic acid that guides protein construction, The octopus ensures that these processes continue to function smoothly, even in temperatures that would impair many other marine animals. The ecological advantages of this adaptation become clear when considering the wide range of environments inhabited by octopuses. Some species live in warm tropical reefs, while others occupy cold temperate waters, or even the dark depths of the deep sea. Each environment presents unique thermal challenges. Instead of evolving entirely new genetic codes for each habitat, octopuses rely on ribonucleic acid editing to adjust their molecular machinery to local conditions. This flexibility may also contribute to the remarkable intelligence observed in these animals. Octopuses display complex behaviors including problem-solving, tool use, and the ability to navigate intricate environments. Their nervous systems contain hundreds of millions of neurons, rivaling the complexity of some vertebrate brains. Maintaining such cognitive abilities across varying temperatures requires molecular systems capable of preserving the speed and accuracy of neural communication. As evening currents begin to flow across the seabed, the octopus retreats toward a sheltered hollow beneath a slab of rock. Its skin darkens slightly, blending with the shadows that gather as sunlight fades above. Within the animal's cells, ribonucleic acid editing continues its silent work, reshaping the instructions that determine how proteins function in the cold water surrounding it. The ocean is a place of relentless environmental change. Temperatures fluctuate with shifting currents. Seasonal cycles alter the chemistry of the water. Animals that cannot adapt to these variations often find themselves restricted to narrow ecological niches. The octopus, however, carries within its cells a molecular toolkit capable of adjusting its physiology with remarkable precision. This ability represents one of the most sophisticated examples of biological flexibility known in the animal kingdom. While many species rely on slow evolutionary change to adapt to new conditions, the octopus modifies the instructions used by its cells in real time. Through ribonucleic acid editing, it transforms its proteins, ensuring that its nervous system remains responsive even in near-freezing waters. In the quiet darkness beneath the rock ledge, the octopus settles into a resting posture. Its arms coil gently around its body, and its breathing slows as water flows steadily across its gills. Outside, the cold ocean continues its endless movement, currents shifting across the seafloor and carrying with them the microscopic signals of life and opportunity. Hidden within the animal's cells, however, lies a remarkable evolutionary innovation. By rewriting portions of its genetic instructions as they are used, the octopus maintains control of its nervous system in environments that challenge the limits of biological function. It is a creature that does not simply endure the cold. Instead, it edits its own molecular machinery to meet the demands of its surroundings, transforming genetic information into a dynamic tool for survival. Here in the cold twilight of the ocean floor, where temperatures test the limits of life, the octopus demonstrates that evolution's most powerful strategies are not always visible in anatomy alone. Sometimes they exist in the invisible chemistry of the cell, where molecules are rewritten and reshaped so that an animal's mind can continue to think clearly, even in the coldest corners of the sea. Along the shifting margins where sea meets land, the tide reveals a landscape that changes character every few hours. At low tide, the ocean retreats, exposing a mosaic of sand flats, rocky ledges and shallow tide pools where sunlight warms the water trapped in small depressions. Barnacles cling to stones like clusters of rough white beads. Snails graze on thin films of algae. Small fish dart nervously through narrow channels, retreating deeper whenever shadows pass overhead. Beneath this busy surface layer of coastal life moves one of the shoreline's most persistent inhabitants, a creature armored in rigid plates and jointed limbs that seem built for durability. A crab emerges cautiously from beneath a slab of rock, its legs moving sideways across the sand in quick, deliberate steps. The animal pauses frequently, lifting its stalked eyes above the sediment as if surveying the surrounding terrain for threats. Its shell, technically known as a carapace, gleams slightly in the filtered sunlight that passes through shallow water. This shell is more than decoration. It is a hardened exoskeleton composed largely of chitin, strengthened with calcium carbonate, forming a protective armor that shields the crab's soft internal organs from predators and environmental hazards. For much of the crab's life, this rigid armor defines its identity as a resilient survivor in one of the ocean's most unpredictable environments. The exoskeleton protects the crab from crushing jaws, abrasive sediments, and the constant turbulence of waves and currents. Yet the very structure that ensures the crab's survival also creates a profound biological dilemma. Unlike animals with internal skeletons, whose bones grow gradually as their bodies expand, a crab's exoskeleton cannot stretch or grow once it has hardened. As the crab's body increases in size, its shell becomes an increasingly restrictive prison. To solve this problem, crabs undergo a process known as molting, in which the entire exoskeleton is shed and replaced with a new, larger one. This transformation is among the most vulnerable periods in the crab's life, a moment when the protective armor that normally shields it disappears completely. The process begins long before the old shell splits open. Inside the crab's body, a new soft exoskeleton forms beneath the existing one, gradually separating from the rigid outer layer through the secretion of specialized enzymes. These enzymes dissolve portions of the inner shell, creating a thin gap between the old exoskeleton and the developing new one beneath it. The crab's body begins to reabsorb valuable minerals, such as calcium from the old shell, storing them internally so they can later be redeposited into the new exoskeleton. This recycling process ensures that essential materials are not wasted, especially in environments where mineral availability may fluctuate. Eventually, the crab reaches a point where the old shell becomes too tight to contain its growing body. Internal pressure builds as the animal takes in water and expands slightly within the confined space. At a critical moment, the exoskeleton splits along predetermined seams, usually across the back of the carapace. The crab begins the delicate task of extracting itself from its former armor. What follows is an extraordinary display of biological precision. The crab pulls its body free from the rigid shell piece by piece, carefully withdrawing each leg, antenna, and eye stalk from its previous casing. The process can take several minutes or even longer, depending on the species and environmental conditions. Once the crab emerges completely, the empty shell remains behind like a ghostly replica of the animal that once inhabited it. At this stage, the crab is no longer the armored survivor that predators fear. Instead, it becomes something entirely different. A soft-bodied creature whose tissues lack the rigid protection that once shielded it from danger. The new exoskeleton beneath the old one is still flexible and delicate, offering little resistance to crushing forces. For a brief but critical period, the crab exists in what might be described as a biological state of extreme vulnerability. The transformation does not end with the shedding of the old shell. In fact, the most remarkable phase of molting begins immediately afterward. With its new exoskeleton still soft and pliable, the crab begins to pump large volumes of water into its body. This intake causes the animal to expand dramatically, stretching the flexible new shell outward to a larger size before it hardens. This process effectively transforms the crab into something resembling a living balloon. Its tissues swell with fluid, pushing against the boundaries of the new exoskeleton. The expansion ensures that when the shell eventually hardens, it will provide sufficient space for the crab's enlarged body. Without this inflation stage, the new shell would remain too small, forcing the crab to molt again prematurely. The swelling can be dramatic. Within a relatively short period, the crab's body increases significantly in size as water fills internal cavities and tissues stretch to their temporary limits. The exoskeleton responds by expanding outward, accommodating the increased volume like a flexible mold, shaping itself around the animal's inflated form. During this phase, the crab must remain relatively still, conserving energy and minimizing movement that could damage its fragile shell. Predators roaming the shoreline, however, are well aware of this vulnerable stage. Fish, birds, and larger crustaceans often search for freshly molted crabs, recognizing that the normally formidable armor has temporarily vanished. To survive this dangerous interval, many crabs seek refuge in concealed locations before molting begins. They retreat beneath rocks, bury themselves in sand, or hide deep within crevices where predators cannot easily reach them. Even with these precautions, the risk remains substantial. A soft-shelled crab caught in open water becomes an easy meal for nearly any predator capable of crushing its delicate body. The period of greatest danger typically lasts about 2 days, a window of time during which the new exoskeleton gradually hardens, as minerals previously stored within the crab's body are redeposited into the shell structure. Calcium carbonate crystallizes within the chitin framework, transforming the flexible outer layer into the familiar rigid armor that defines the crab's appearance. During these 2 days, the crab continues to regulate the amount of water within its body, slowly adjusting its internal pressure as the shell begins to stiffen. Excess fluid may be released gradually as the exoskeleton solidifies around the animal's newly expanded shape. Once the shell reaches full hardness, the crab regains its defensive strength and resumes its active role within the ecosystem. The ecological implications of this molting process extend beyond the individual crab. In many coastal environments, molting events occur throughout the year, creating brief windows when normally protected crustaceans become vulnerable prey. Predators that specialize in crushing hard shells may shift their hunting behavior to target recently molted individuals, taking advantage of this temporary weakness. At the same time, the molting cycle contributes to the dynamic recycling of nutrients within marine ecosystems. The discarded exoskeletons left behind by molting crabs eventually break down, releasing calcium and other minerals back into the environment. Small scavengers feed on fragments of these shells, incorporating their nutrients into the food web. For the crab itself, molting represents both a necessity and a gamble. Growth cannot occur without shedding the old exoskeleton, yet every molt exposes the animal to extraordinary risk. Some species molt multiple times during their lives, gradually increasing in size with each new shell. Others molt less frequently as they mature, eventually reaching a stage where growth slows and molting events become rare. The process also influences social interactions among crabs. In species where territorial disputes or mating competitions occur, individuals may time their molting periods carefully to avoid conflicts while their shells remain soft. Some crabs even synchronize molting with environmental cues, such as tidal cycles or seasonal changes, ensuring that the vulnerable stage coincides with conditions that reduce predator activity. As the tide begins to rise again along the shoreline, water floods back into the pools and channels carved into the sand. A recently molted crab lies hidden beneath a flat stone, its body still swollen with the water it pumped inside after escaping its previous shell. Its new exoskeleton remains soft to the touch, flexible enough that the crab must move cautiously to avoid damaging the delicate structure. For the moment, the animal resembles a fragile version of its armored self, a creature that has temporarily abandoned the protective fortress that normally defines its identity. Yet this vulnerability is only temporary. Within the tissues of the new exoskeleton, minerals are slowly crystallizing, transforming the flexible framework into a hardened shield capable of withstanding the pressures of life along the coast. Soon, the crab will emerge once more from its hiding place, its armor restored, and its body slightly larger than before. The risks of molting will fade into memory until the cycle begins again in the future. In the meantime, the crab resumes its role as both hunter and scavenger, navigating the constantly shifting terrain of the shoreline with the same resilience that has allowed crustaceans to thrive for hundreds of millions of years. Here, in the restless boundary between land and sea, survival often depends on timing, adaptation, and the willingness to endure moments of extreme vulnerability. The molting crab embodies all three, transforming itself into a temporary creature of soft flesh and liquid expansion before reclaiming the armored identity that defines its place in the marine world. Far beyond the protective edges of coral reefs and coastal shallows, the open ocean stretches into a vast moving wilderness where survival depends not only on strength, speed, or biological resilience, but increasingly on memory. Here, the sea is not a stable environment. Currents shift across thousands of kilometers. Temperatures fluctuate as climate patterns change. Fish populations rise and collapse with the rhythms of ocean cycles that may take decades to complete. For many marine animals, these fluctuations represent existential threats. Entire populations can disappear when prey migrates away, or when warming waters disrupt delicate ecological balances. Yet within this unpredictable realm lives a predator whose greatest defense is not armor, venom, or biochemical adaptation, but knowledge passed down through generations. The ocean's largest member of the dolphin family moves through these waters with calm authority, its black and white body slicing effortlessly through rolling swells. A dorsal fin rises high above the surface, cutting a sharp silhouette against the horizon before disappearing again beneath the waves. The animal is an orca, often called a killer whale, though in reality it is neither a whale nor merely a hunter. It is a carrier of cultural memory, a living archive of survival strategies accumulated across decades. The pod travels together in a loose formation, their movements coordinated with subtle precision. Several individuals surface in rhythmic intervals, releasing bursts of vapor into the cool air before slipping back beneath the surface. Beneath the water, their bodies glide through the deep blue in synchronized arcs, each animal adjusting its pace and direction to maintain the cohesion of the group. This social structure defines nearly every aspect of an orca's life. Unlike many predators that disperse after reaching maturity, orcas remain bound to their family groups for life. At the center of this family structure stands a figure whose importance extends far beyond simple leadership. The matriarch, often the oldest female in the pod, carries within her mind an extraordinary record of environmental knowledge. Her lifespan can exceed half a century, sometimes stretching even longer. During that time, she experiences shifts in climate patterns, fluctuations in prey populations, and changes in migration routes that younger animals have never witnessed. Each event becomes part of a memory bank that guides the survival of the entire pod. In the open ocean, where food resources can be scattered across immense distances, knowing where to hunt is often more important than physical strength. Salmon runs move along specific coastal pathways that may change subtly over time. Seals and sea lions gather at seasonal breeding sites known only to predators that have learned their locations. Squid migrate vertically through the water column in nightly patterns that shift with lunar cycles and ocean temperatures. A pod without access to such knowledge risks wandering through barren waters while other groups thrive. The matriarch functions as a living map of the ocean's hidden resources. Decades of experience allow her to recognize subtle environmental cues that signal where prey may gather. Changes in water temperature, variations in salinity, or even the behavior of seabirds overhead can reveal information about the distribution of fish below the surface. Younger members of the pod observe and follow these decisions, gradually learning the complex geography of hunting grounds that cannot be seen on any chart. The importance of this cultural knowledge becomes especially clear during periods of ecological upheaval. Climate shifts, whether driven by natural cycles or human-induced warming, can dramatically alter the distribution of marine life. Fish populations that once thrived in certain regions may migrate toward cooler waters. Ice patterns in polar seas may change, affecting the availability of seals or whales that serve as prey. For species that rely solely on instinct, such disruptions can lead to starvation and population collapse. Orca pods, however, possess an additional layer of resilience. The matriarch's memory may include routes to distant feeding grounds that become critical when local resources decline. She may recall a salmon run that occurred decades earlier during a similar environmental pattern, or a deep offshore canyon where prey once gathered during a period of warm water. These recollections guide the pod toward alternative hunting grounds when familiar locations fail to provide sufficient food. Scientific studies of certain orca populations have revealed striking evidence of this phenomenon. Pods led by older females often experience higher survival rates during years when prey becomes scarce. Younger groups lacking an experienced matriarch may struggle to locate adequate food, while pods guided by a memory-rich leader navigate directly toward productive hunting areas. The difference between survival and starvation can hinge on knowledge accumulated over half a century. As the pod travels through the open water, the orcas maintain constant communication through a series of vocalizations that echo across the ocean. These sounds range from sharp clicks used for echolocation to complex calls that serve social and navigational functions. Each pod possesses a distinctive set of vocal patterns, a dialect unique to that family group. These acoustic traditions are passed down through generations, reinforcing the social bonds that hold the pod together. Echolocation allows the orcas to perceive their environment with remarkable precision. By emitting bursts of sound and analyzing the returning echoes, they can detect fish schools, underwater terrain, and even the movements of individual animals hidden in darkness or murky water. The matriarch's experience helps interpret this acoustic landscape, guiding the pod toward prey concentrations that might otherwise remain undetected. On this particular day, the pod approaches a coastal region where cold, nutrient-rich currents meet warmer offshore waters. Such convergence zones often attract large schools of fish, creating feeding opportunities for predators capable of locating them. The matriarch slows her pace slightly, adjusting her course with subtle movements of her tail. Beneath the surface, her echolocation clicks pulse through the water, scanning the depths ahead. Moments later, the pod changes formation. Several individuals spread outward, forming a loose semicircle around an area where dense echoes reveal the presence of a large salmon school. The fish move tightly together in a defensive cluster, their bodies flashing silver as they twist through the water. For smaller predators, this formation would be difficult to penetrate. For the coordinated hunters of an orca pod, it represents an opportunity. The attack unfolds with practiced efficiency. Younger orcas drive the fish toward the center of the formation, while others approach from below, disrupting the school's cohesion. The matriarch remains slightly behind the main action, observing the movements and adjusting the pod's strategy through subtle shifts in direction and bursts of vocal communication. Within minutes, the tightly packed school begins to fragment, individual fish breaking away from the protective cluster. Each orca seizes prey with powerful jaws capable of crushing bone and muscle alike. Yet even during the feeding frenzy, the structure of the pod remains intact. Calves stay close to their mothers, learning through observation how the adults coordinate their movements during complex hunts. These experiences form the foundation of the cultural knowledge that will eventually guide the next generation. As the feeding concludes, the pod regroups and resumes its steady journey along the coastline. The ocean around them continues its slow transformation under the influence of changing climate patterns. Warming waters alter the distribution of plankton, which in turn affects the populations of fish that depend on these microscopic organisms. Some species migrate to cooler regions, while others decline entirely. In such a shifting environment, the orca's reliance on cultural transmission becomes even more important. Each generation inherits not only genetic adaptations, but also behavioral traditions shaped by decades of accumulated experience. The matriarch's memory functions as a stabilizing force within this dynamic ecosystem, guiding the pod through environmental challenges that would overwhelm less socially complex predators. The significance of this cultural fortress becomes particularly evident during extreme climatic events. Marine heat waves, for example, can disrupt food webs across vast areas of ocean, reducing prey availability for many species. During such periods, some marine predators experience dramatic population declines. Orca pods led by experienced matriarchs, however, often adjust their movements to exploit alternative prey or previously used feeding grounds. This adaptability reflects a deeper truth about the nature of survival in complex ecosystems. Biological resilience is not always a matter of physical toughness or chemical defenses. Sometimes it depends on the ability to remember, to learn from past experiences, and to share that knowledge across generations. In the case of the orca, memory itself becomes a survival mechanism as powerful as any evolutionary adaptation. As evening approaches, the ocean surface darkens under the soft glow of a descending sun. The pod continues its journey through the coastal waters, their dorsal fins cutting graceful arcs across the surface. Somewhere ahead lies another feeding ground known only to those who have traveled these routes for decades. The matriarch leads with quiet confidence, guided by memories formed long before many members of the pod were born. Within her mind exists a map of the ocean that no satellite image could fully capture. It includes not only the locations of prey, but also the timing of migrations, the subtle shifts in currents, and the patterns of environmental change that unfold across generations. Each piece of information contributes to a cultural inheritance that protects the pod from the uncertainties of a changing world. In the vast and unpredictable wilderness of the open ocean, the orca survives not merely as a predator, but as a keeper of knowledge. Its strength lies not only in muscular bodies and powerful jaws, but also in the quiet transmission of memory from one generation to the next. Here, Beneath the rolling swells and endless horizons, survival is secured by a fortress built not of bone or armor, but of experience, shared learning, and the enduring wisdom of the matriarch who remembers where the ocean once fed her family and knows where it will feed them again.